Okay. So we can start.
Sure, sir.
Yeah. So I welcome all of you in inaugural ceremony of the 15th Indo-Malaysian Conference on global progress and upcoming challenges in pharmaceutical sciences. Today on 11th March, it will be a two-day event, 11th and 12th March. 11th March will be the online sessions by Dr. Paramita Basu from Toro College of Pharmacy, USA, as well as Dr. Sivkanna Filoria from AIMST University, Malaysia. So, in the inaugural ceremony, there will be welcome speech by Professor S. Sankar, who is member IQAC at Vijaya Institute of Pharmaceutical Sciences for Women and advisor IQAC Cluster, India, and retired principal and advisor at ANR College, Gudalwada. I'm pronouncing correct, I think, Andhra. We have another guest, Dr. Vetri Selvan Subramaniam, who is Vice President of APP Malaysian International Branch and Associate Professor at Department of Pharmacology, Mehsa University, Kuala Lumpur, Malaysia. Welcome, sir.
Thank you.
We
Thank
have
you,
Dr. Sunita Dahiya, who is General Secretary of the Association of Pharmacy Professionals and Organizing Chairman and Assistant Professor at School of Pharmacy, University of Puerto Rico, San Juan, Puerto Rico, USA. Welcome, Dr. Dahiya. And as earlier said, there will be two online lectures today by Dr. Paramita Basu from Toro College of Pharmacy, New York, USA, as well as Dr. Sivkanna Filoria from AIMST University, Bidong, Kedah, Malaysia. The chairpersons for the today event are Professor Piyush Mittal, who is Professor of Pharmacy Practice at Tirthankar Mahavir College of Pharmacy at Tirthankar Mahavir University, Moradabad, Uttar Pradesh. Welcome, Professor Mittal. We have Professor Somnath D, who is Professor of Pharmacology at St. Paul College of Pharmacy, Turkamjal, Rangareddy, Telangana. Welcome, Professor Somnath. So, Professor Mittal and Professor Somnath both will be the chairpersons of the today invited lectures. These will be followed by the oral presentation. There will be oral competition today, where 8 research scholars will deliver that 15 minutes orations, and the awardee will be subjected to the Sri Tarsemjan Memorial Award for Outstanding Oration. And at last, there will be award announcement, which will be by me between 4 to 5 p.m. So, and that will conclude the today's session, and 12th March session will be total offline and will be conducted by Professor Padmalatha. So, as part of inaugural ceremony, I invite Professor Sankar for the welcome speech. Professor Sankar, please.
Thank you, sir. I deem it a privilege to deliver the opening remarks and the welcome speech on behalf of the Internal Quality Assurance Cell of VIPW, Vijaya Institute of Pharmaceutical Sciences for Women. And I welcome you all to the 15th Indo-Malaysian Conference on Global Progress and Upcoming Challenges in Pharmaceutical Sciences. This conference, you are all aware, is being organized in collaboration with APP Malaysian International Branch and APP Molecular Farm Division. First, a few words of introduction about the host institute, VIPW, Vijaya Institute of Pharmaceutical Sciences for Women. This college was established in 2009, 13 years back, by SRK Foundation under the chairmanship of Boypati Srinivasa Apparao. If you look at Padmalatha madam's screen, there will be a person standing, you see, in a poster behind her. You can visualize that person, a dynamic engineer who has been associated with various colleges, the Siddhartha Academy, and particularly SRK Foundation, SRK College Engineering, and this VIPW. His son, the dynamic Sri B.S. Krishna, with the assistance of our own principal, Dr. K. Padmalata, has been looking after the day-to-day administration of the college. A college cannot survive, you see, unless it has dedicated staff, and we have a lot of dedicated personnel serving the needs of the students and serving the cause of education. And we are happy to announce that recently the National Accreditation Council, NAC, Assessment and Accreditation Council of India, with its headquarters in Bangalore, has assessed this college and gave it A+ grade, perhaps the only college in the whole of India to receive this kind of accreditation status. It has been possible only because of the concerted effort of all the stakeholders and the dynamic leadership of Professor Padmalata, the management secretary S.R. Krishna, and then the other faculty members and the student friends too. The institution has been offering, you see, various programs, B Pharmacy, Pharma D, M Pharmacy, with several combinations. And the vision of the college is to become a potential leader in the field of pharmacy through excellence in education and research. And the grade reiterates the college's commitment to excellence. The goal of Vijay Institute of Pharmaceutical Sciences for Women is to empower girl students, to develop a positive attitude in them, to promote academic excellence, to build competencies, and also to support the economically challenged, and to make them employable by improving their employability skills. And at the same time, you see, as social training, it wishes to foster integrity in the students, and thus it has created a brand image for itself. There is an academic environment in the college, you see, a campus culture where all students acknowledge and bring out their potential based on ethical conduct. To ensure the optimum usage of infrastructure and other amenities like library, laboratories, and IT facilities, the college has been working without any rest, relaxation, to impart a learner-centric and inquiry-based instruction. And this is something I just wish to present as a background note, because this is the host college organizing this two-day webinar and face-to-face interaction scheduled for tomorrow. Going back to the topic on hand, pharmaceuticals have been used to treat illnesses for thousands of years. The early days of medication included the use of plants and herbal remedies to treat a variety of diseases and traumas. In fact, you see, Ayurveda, which originated in India, is perhaps as old as 3,000 years ago, has a long tradition behind it. Even now, it remains a favored form of healthcare in large parts of Eastern, especially in India, where a large percentage of the population uses this system exclusively or combined with modern medicine. Today, the long and complex journey to demonstrate the safety and efficacy of a compound and bring it from the laboratory into the hands of patients in need is a multi-billion-dollar global pharma industry. Pharmaceutical companies world over have been continually striving towards innovative new treatments that help people live longer and, of course, healthier lives. And their therapies are developed, are manufactured and marketed and distributed around the world by various pharmaceutical companies every day. It's hoped that this conference will enable the participants to look at some of the key contributions of the industry and the reasons why pharmaceutical companies are so important to patients and to the society and to the life sciences industry. because this industry has greatly contributed to the increase of life expectancy for people across the world. In the early 20th century, up to perhaps between 1901 and 1910, the global life expectancy was, as scientific journals reported, only 32 years. Now it rose to 72 years. This has been possible only because of the pharmaceutical innovations, which did not just benefit richer nations, but they also helped developing countries and positively impacted global inequality in life expectancy. Life expectancy is increasing, and the problems of individuals because of diseases is coming to a lower level. Pharmaceutical industry strives to eradicate and eliminate diseases. Disease eradication is the ultimate goal while developing treatments, as this benefits ecosystems on a global level. To date, the WHO, World Health Organization, has declared smallpox as the first and so far only human disease to be eradicated globally. And let's hope that in future the pharmaceutical industry will be able to come up with remedies, vaccines, which would eradicate other diseases also. Pharmaceutical industry has been responsible for reducing the pain and suffering of patients by providing treatments to manage pain, symptoms, and side effects. Pharmaceutical companies can improve patients' quality of life, allowing him or her the freedom to live healthier and more fulfilled lives. By preventing diseases, the vaccines produced by pharmaceutical industry, they not only help to save millions of lives, they save money too. Once again, I wish to quote WHO. For every $1 the US spends on childhood vaccinations, over $10 in disease and treatment costs is saved, as per a report of WHO. Moreover, you see, because of the contribution of pharmaceutical industry, the stay in hospital, the time of stay in hospitals has come down. 50 years ago, the average hospital stay for any problem was around 8 days. Now it has come down almost to half, say between 4 and 5 days. Pharmaceutical companies are also responsible for millions of jobs across the world. This is not just in industries. There are academic institutions like our own VIPW and independent laboratories which have been functioning all over the world, which employ a considerable number of people in different countries. Pharmaceutical companies boost the global economy by driving medical progress by researching, developing, and bringing new medicines that improve health and quality of life for patients around the world. So it's a key asset to the global economy. The industry's research and development enterprise drives sizable economic impacts, and the sales of products are at a higher level than any other manufacturing industry. You can see any production you can take for example. Overall, pharmaceutical companies and allied organizations play a pivotal role in helping communities in general and the patients in particular. They provide more than potential cures and life-saving treatments. They also create fulfilling jobs and thus fuel the global economy. Looking ahead, the industry will continue to firmly establish its importance in the world by creating more exciting and groundbreaking treatments. Friends, we are meeting at a time when the COVID-19 pandemic disrupted the delivery of medicines and the other health products worldwide, particularly in low and middle income countries, and exposed the critical gaps in health systems. As new attempts to control the pandemic gathered pace, governments and industry faced new access to medical challenges, not least how to continue delivering healthcare while developing new products for COVID-19 patients and getting them to all corners of the world at unprecedented speed. Further action is required, we all know, to curb this pandemic and prepare for the future by building strong and responsive health systems. As Jayashree K. Iyer said, pharmaceutical companies are not siloed entities with individual supply chains and networks. They have strong relationships of trust, clearly boosted the speed and agility of the industry's response in delivering medicines and vaccines. but for the presence of pharmaceutical industry, see, a bigger percentage of the world's population would have been wiped off with COVID-1, COVID-2, and so on and so forth. We need a better system of crisis preparation and response, we are aware, which means preserving what is currently in place and working together more closely. Let us take a stand to recognize our limitations, complement our strengths through partnerships and downstream collaborations. This conference is itself an example of partnership and collaboration. Let us identify and resolve supply chain challenges, building on pilots and developing new ideas to help better prepare for the next pandemic, to ensure continuity of care and to build local capacity and to address ongoing access challenges. Let us embrace innovations in information sharing to improve supply chain alignments to reach more people. Let us ensure new products quickly reach people in low and income countries, LMICs, by involving generic medicine manufacture early in the process. We have in our own college, we have a generic medicine counter also. Perhaps see members who attend the college VIPW tomorrow, kindly request them to visit this generic medicine stall. Let us also invest in larger network of partners that can quickly ramp up local manufacturing capacity. Let us build inventory reserves supported by large networks of API suppliers. And lastly, let us plan for the unexpected, including creating protectionism. I would have given the details of the program, but Rajiv sir was very kind enough to give an elaborate statement of what is going to come after this welcome speech. I once again extend you all a warm welcome to the sponsors, to the resource persons, to the participants, organizers, to the management of VIPW on behalf of the stakeholders of Vijaya Institute of Pharmaceutical Sciences for Women. I hope that the insights from this two-day conference will be beneficial to all stakeholders and will also result in fruitful outcomes for the public health. Thank you. Thank you so much for giving me an opportunity to speak on behalf of the IQAC of VIPW. I thank and take leave of you.
Thank you, Professor Shankar, for your valuable words. And so I forgot to mention this particular conference is being organized in commemoration of International Women's Day, which was on 8th of March. And we are honoring a lot of women for their scientific potential during the conference.
Sir, you are not audible.
Okay. Now I'm audible?
We can hear you.
Yes, sir.
So thank you, Professor Shankar, for your valuable words. So I forgot to mention this two-day conference we are here we are celebrating the International Women's Day, which was on 8th of March. And through this day, we are honoring a lot of women for their scientific potential, as will be there in the award announcement section. So, Professor Sundar is with us.
Yes, sir.
Yeah, so now I will invite Professor Sundar to read the academic achievement of Dr. Vetriselvan, and then Dr. Vetriselvan will proceed for the his welcome. Professor Sundar, please.
Yes, sir. Thank you, sir. It is my great pleasure to introduce today's speaker, Dr. Paramita Basu. Dr. Paramita Basu is an associate professor of biomedical sciences.
Professor Sundar, you need to continue with Dr. Battery Selvan.
Yes, sir. Okay, sir. Thank you.
Professor, Prof Sundar, shall I give
sir,
my short speech, or you can continue with the speaker?
No,
now we
he
are
has
going to
to
proceed, sir.
introduce you. He
Okay.
has to introduce you.
Dr. Paramita Basu is associate professor of biomedical sciences in the Department of Pharmaceutical and Biomedical Sciences.
Professor Sundar, that will come
Yes, sir.
after. Okay, Dr. Battery, you can proceed.
I can myself. I can give the introduction.
Okay.
Sorry, sir.
Go ahead,
With the veterinary Subramanian, sir.
Thank you, Prof. Sundar, and thank you, the President APP.
Just a minute.
I congratulate all women
Just a minute, sir.
Please unmute your mic. Too noisy. Naga Durga. Yes.
Firstly, I congratulate all the women for now they're celebrating their day, for their courage and their faith. It's very important for us. In every family, is how the women are very important. We all knows. Okay, once again, I congratulate all women. Happy Women's Day. So I thank to the President APP, Prof. Rajudia, for inviting me this wonderful conference. I thank to the management and the organizing committee, Vijaya Institute of Pharmaceutical Sciences and APP 15th Indo-Malaysian Conference. I'm very happy to participate this event. Myself, my background is a pharmacy. Currently, I am the Deputy Dean of Faculty of Medicine, Bioscience and Nursing. Even though I feel very comfortable this event, I feel that this is our conference. My route from pharmacy, but I'm currently I'm working in Faculty of Medicine. When Prof. Rajudia he told me to attend this event. I'm very happy to attend this event. Also, I need to share my experience of this association. After I have joined this association in around 2015. I am very happy to. I have been many places in India. I have participated. I have delivered my speech in different topics. When we had a discussion with all pharmacy professionals, one thing I learned from the knowledge sharing of knowledge and how to develop our field, how to improve our personal developments, not only the personal developments, our skills and the future developments, how we can go for it with the current and future the aim of our field. So many things we had a discussions. With this platform, I feel that is very important for us. Every pharmacy person have to feel very proud of this association, because this is the platform to meet each other. We can share our knowledge on the current field and the future. What we are going to do in the society is very important. So with that, I'm very happy to attending this conference. Also, I'm waiting for their speaker, how they're going to deliver their topics, especially Dr. Paramita, who is going to give a speech. Paramita is working in Tauro College of Pharmacy in USA. And one more speaker I need to introduce, Prof. Shivakanya Floria, who is working in Ames University. I know Prof. Shivakanya Floria personally. She's a dedicated and long services in pharmacy society. Apart from that, I congratulate all pharmacy professionals, those who are attending here. This event, I hope this event is a fruitful event. End of the day, we used to feel something we learned from the conference. Also, in the future, we need to get some more idea how to proceed in our future progressions. So these are all very important for us as a health, as a pharmacy profession, as a pharmacy background person. I'm telling, I wish you all the very best for all of you to go to move forward the next level. All the very best for to everyone. Thank you very much.
Thank you, Dr. Vetri Selvan,
Thank you
for the welcome speech. So, Dr. Vetri Selvan is our Vice President for the APP Malaysian International Branch, and who is very active member. And we have organized many events, keeping him as speaker, and he's always very supportive in all the conferences and other related activities. Thank you, Dr. Vetri Selvan.
Thank you very much
For your time.
Thank
So now I will ask the General Secretary, Dr. Sunita Dahiya, who is organizing chairman of the event also, and who is presently the assistant professor at Department of Pharmaceutical Sciences at School of Pharmacy, University of Puerto Rico, San Juan, USA, for the opening remarks. Dr. Dahiya, please. Dr. Sunita Dahiya.
Oh, I'm sorry. Can you hear me now? Okay.
Yes, you can go ahead.
Okay, thank you. A very good morning to all. On the occasion of the International Women's Day on the 8th March, I am delighted to welcome all of you in the APP 15th Indo-Malaysian Hybrid Conference hosted by Vijaya Institute of Pharmaceutical Sciences for Women, Vijayawada, Andhra Pradesh. The theme of the conference is Global Progress and Upcoming Challenges in Pharmaceutical Sciences. For today's virtual component of this conference, we have two renowned speakers with us. The first speaker is Dr. Paramita Basu from Toro College of Pharmacy, New York, USA, and she will be speaking on characterization of penicillin tolerance in Group B Streptococcus. Our second speaker for today's event is Dr. Shivkanya Fuloria from AIMST University, Malaysia, and she will be speaking on endophytes, the important source of bioactive molecules. After the lunch break, the conference will proceed with e-poster presentations and orations under late Shri Tarsim Jain Memorial Oration Competition 2022, for which we have selected 8 oral presentations, and each participant will be presenting on their research and sharing their knowledge with the attendees. Also, the association is pleased to celebrate the International Women's Day with our all women speakers and participants in oral and poster sessions under this platform of Indo-Malaysian Conference. On behalf of Association of Pharmacy Professionals, APP Malaysian International Branch, and APP Moldsoft Division, I express my sincere gratitude to Shri B. S. Sri Krishna, Patron and Secretary, SRK Foundation, Vijayawada, and Professor K. Padmalata, Convener and Principal, Vijaya Institute of Pharmaceutical Sciences for Women, to host this event in commemoration of International Women's Day. I also express my appreciation to Professor S. Sankar, who is member IQAC VIPW and advisor IQAC Cluster India, and Dr. Vetri Selvan Subramaniam, who is Vice President APP Malaysia International Mahasa University, Kuala Lumpur, Malaysia. Professor Rajita Galla, Co-Convener and Vice President, APP Andhra Pradesh State Branch and Institute of Pharmaceutical Technology, Sri Padmavati Mahila Vishwavidyalayam, Tirupati. Dr. Madhu Gupta, Scientific Committee Co-Chairman, School of Pharmaceutical Sciences, Delhi Pharmaceutical Sciences and Research University, New Delhi. Dr. Gaurav Gupta, Organizing Secretary, National Head, APP Molfarm Branch, School of Pharmaceutical Sciences at Jaipur National University, Jaipur, Rajasthan. Professor Piyush Mittal, Department of Pharmacy Practice, Tirthankar Mahavir College of Pharmacy, Tirthankar Mahavir University, Moradabad, Uttar Pradesh. Professor Suresh V. Chennupati, Department of Pharmaceutical Analysis, Nalanda College of Pharmacy, Charlapalli, Nalgonda, Telangana. Professor Anju Goyal, Department of Pharmaceutical Chemistry, BN Institute of Pharmaceutical Sciences, Bhopal Nobel University, Udaipur, Rajasthan. And along with all this personnel, I specifically mentioned the organizing committee members of this committee, the management of the Vijaya Institute of Pharmaceutical Sciences for Women, also the APP office bearers, and of course, the attendees of this conference. With these words, I again welcome and thank all the participants and attendees to make this event successful. Thank you very much.
Thank you, Dr. Daya, for your valuable words and opening remarks. So now, before not taking much time, we will go for the scientific sessions, which will be chaired by the Professor Mittal and Professor Somnath, who are professors of pharmacy practice. Professor Mittal is Professor of Pharmacy Practice at Tirthankar Mahavir University, Moradabad, and Professor Somnath is Professor of Pharmacology at St. Paul College of Pharmacy, Turkamjal, Telangana. So they will lead the session as the chairperson. Now I will invite Professor Sundar to say a few words about Dr. Basu, who is our first speaker today.
Thank you, sir. This is my great pleasure to introduce today's speaker, Dr. Paramita Basu. Dr. Paramita Basu is an associate professor of biomedical sciences in the Department of Pharmaceutical and Biomedical Sciences in Toro College of Pharmacy, New York, USA. She is a teaching of PharmD, MD, and nursing students. She is also an infectious disease researcher with a passion for public health and community medicine, as well as experience in biotech and pharmaceutical drug design and development. She earned her MS and PhD in biological sciences from St. John's University, New York, USA, and MTech Biotech from Jadavpur University, India. In 2011, she received internal grant from Toro College of Pharmacy Faculty Research Fund. She has authored one book and published a number of research articles in reputed journals and 5 book chapters also. She received Faculty Service Award, Faculty Research Award from Toro College of Pharmacy, and she is a public health web page editor also. So this is the brief introduction about Dr. Paramita Basu. Now I hand over the session to Madam. Madam, please.
Thank you so much for the introduction, Dr. Sundar. I guess only, so since the last month, since February, I actually also have a joint appointment in the College of Podiatric Medicine. That's a new development. I am the preclinical director of infectious diseases too. So that's a very new development. So that's another title which I'll probably be adding to my signature soon, but my office is changing. But it's good, like that's probably my first so I'm moving from the College of Pharmacy to the College of Medicine. I do have a basic degree in pharmaceutical biotechnology, so my basic background is more of a biotechnology background. but because my background is more in antimicrobial resistance, so I guess I get to work in both colleges. So I do work, a big part of my research interest is in antimicrobial resistance and in public health, as you already mentioned. Certainly, I look at biofilms as well as penicillin tolerance, which is one of the major causes. I have to share my screen. I need the screen sharing ability. Is it possible? Can I?
You are not the host or co-host?
I am. It's saying it's my screen sharing is disabled.
Professor Padmalata, we need to make her
Raju sir, you will have to share.
Hello?
Yes, sir. You will have to share the screen, sir. You are the host now.
No, you have to make Dr. Basu as co-host. She will share by herself.
Okay, sir.
Can I start sharing? Not yet. It is. Whenever you're ready. Otherwise, I can send this file.
Sir, Raju sir.
Yeah.
You will have to make her co-host.
Okay. Okay. 1 minute. I'm trying. Yes, Dr. Basu, you are the co-host now.
Okay. Let me see if I can share now. Seems like okay, this looks better. Certainly. Okay, can everybody see my screen now?
Yes.
Thank you. Okay, so so this is the work that we have been doing for a little bit of time, and I'm not alone. This is other than me, there I have a couple of this work has been done over the years by a few other students. So I'm presenting the work of Hyen Kim, Bruna Fittipaldi, Calvin Wong, Faria Hoke, and Ricky Lee. They are also my students. Some of them have already graduated. Some of them, Bruna is working for Bruna and Calvin. They're both working for Pfizer, and Hyen and Faria, they are going to graduate. Ricky still has a couple of years to graduate. So this is a work in progress. So the penicillin tolerance is a relatively less known concept, in the sense that molecular mechanism of penicillin tolerance is not very well characterized. There are a couple of possible mechanisms, but again, there is really not a very well-known mechanism that's well established. Our lab is working to identify specific genes to develop PCR-based assays. There are already assays that are used for diagnosing Group B strep for clinical diagnosis, but we want to identify biomarkers, possibly for use for multiplexing that same assay, to add this into the assay as a second biomarker, to also identify penicillin tolerance at the same time while screening for GBS. Usually, our idea is to design fluorescence-based assays for a fast, high throughput measurement of the degree of penicillin tolerance. Group B strep or GBS, it's the number one cause of neonatal infection and mortality in the United States. It causes pneumonia, bacteremia, sepsis, and meningitis in newborns, and usually it causes a fast and quick death in newborns within the first 5 days after birth. Typically, the standard way this is dealt with in the US hospital is by universal screening of all women at 35 to 37 weeks of gestation. This screening has started since 2002 by the recommendations of CDC. And since this screening started, there was a sharp decline in the number of GBS deaths of newborns. But even then, the ones that do happen, since there is no report of the standard treatment is penicillin. So since there is no reported resistance, we think of tolerance as one of the causes. Typically, it's the mother, the pregnant woman, is asymptomatic during pregnancy. So we think that the GBS is carried maternally in 12 to 28% of the pregnancy cases, and neonatal colonization happens during labor and delivery in 30 to 50% of the cases, and this leads to about early onset neonatal GBS disease, and this takes place when the baby is passing during the birthing process through the birth canal in less than 1%. And of course, any neonatal colonization is very dangerous for the baby. It is also a major cause of maternal uterine infection and septicemia, but it is treatable. But for babies, it's difficult to treat. And this is why it's very important to be able to diagnose it right in the beginning. Possibly, it's important to diagnose if the infection is going to be treated by penicillin or not right in the beginning, possibly even before the baby is born, because every day is really important for this treatment for the baby. Also, it's a cause of skin and soft tissue infections in elderly and diabetics. Typically, the first line of treatment is penicillin G for GBS. For people who are allergic to penicillin, the second line of treatment is erythromycin or clindamycin, and then the last option is vancomycin. For penicillin, a model of penicillin tolerance is of course different from penicillin resistance, which is really not being reported. And penicillin tolerance is thought of being the leading cause of neonatal infection, particularly since the 2002 introduction of the screening protocol, which, as I told you already, lowered the number of neonatal deaths by significantly. And the idea of tolerance versus resistance is that when there is an infection with GBS, you can see that the GBS titer in the blood goes up, and then at the point where the penicillin is added, which is this point in time, the growth starts going down. It kind of stays down, but as you can see, it's not zero. Simply there is a growth inhibition, but the cells remain viable. So basically, it becomes so low that it remains undetectable, but even by a PCR-based test. Once the physician thinks that the infection is cleared, the typically the penicillin is given 1 dose of penicillin once the treatment is withdrawn. In a tolerance situation, typically the GBS starts growing again. However, in a resistant, if it was, there is no resistance reported, but in a resistant infection, it would have kept on growing. But there is no resistance. We are talking about the blue curve here. If it was susceptible, it would have died. But in a tolerant reaction, it keeps on laying low, and then once it is withdrawn, the antibiotic is withdrawn, it starts growing. That is what I mean. That is how we define tolerance. And with the viability, it's the same thing. With viability, the time it goes down. In case of tolerance, as you can see, it kind of goes slowly over time. Resistance is defined as something that's not affected by an antibiotic at all. So there are a couple of mechanisms of by which penicillin, goes through its bactericidal effect. Some of the mechanisms that are reported in literature, as you probably already know, are through penicillin binding proteins, or PBPs, and that's one model. And then the other model is induction of autolysis of the bacteria. And there are a couple of, there's quite a number of different, looking at a review of literature, we were able to identify a lot of reports of penicillin tolerance. We weren't the only groups, just nothing much about the mechanism. So, and because we couldn't really find anything in group B strep, we had to look for models in, say, group A strep, like Streptococcus pneumonia and Staph aureus, and that's where we started looking for the molecules or the proteins. So the penicillin, to give you like a summary of what were the kinds of models that we were finding in literature. So we found two basic groups. One was that there was a difference in these tolerant strains. There was a difference in their cell wall structure. Their cell wall was physically different. It was constructed differently. It was in terms of thickness or cross-linking, and their rate of synthesis. It had a faster rate of the synthesis of the cell wall. That's why it was stronger. And then the other groups of papers, they had said that there was a difference in the signal transduction, and these fell under the autolysin regulation model. Usually, these were seen in Staph aureus. And these involve the choline, anti-choline proteins and their regulation. And then in pneumococcal models, these involved stress responses using the CIA-HR two-component histidine kinase signal transduction mechanisms. It kind of involves penicillin, which binds with the PBP, inhibiting cell wall synthesis and cell division, overexpressing CDCIDA and CIDP. I don't want to go into the details. And also the others are the autolysin model, where it basically bores a hole into the membrane, and that basically decreases the cell membrane potential, and that's how changes the membrane potential, and so on and so forth. So our plan of action, basically, of going about this project, was to first identify the specific proteins. We go up or down in response to penicillin treatment. We really didn't know what were we looking for. So it was a fishing expedition. So we really wanted to go for an RT-PCR to look at which were the proteins that are changing once we subjected to penicillin. So we had to do RT-PCR study. We were looking at CIA-H, CIA-R. We found nothing really. And then we also started looking for membrane permeabilization. So we started looking at fluorescence staining. We used redox sensor green and propydium iodide staining to look for degree of viability and membrane permeability before and after penicillin treatment. We also looked at fluorescence activated cell sorting based assays to measure more of a quantitative measure of viability of membrane lysis. And then we looked at cell wall construction, particularly with respect to penicillin binding proteins between the susceptible strain and the penicillin tolerant strains. And then in the future, then the next group of targets would be to knock out and overexpress the genetic targets of the choline anti-choline model proteins. That's something that we plan to do that's the next steps. So first, of course, we established what are definitions of tolerance. And to do that, we use the MIC90 and MBC90 ratio. So we were looking at minimum bactericidal concentration and minimum inhibitory concentration, where we were looking at where at least more than 90% of the cells became non-viable. Bactericidal being if they were able to remain viable after growing them separately again on plates. And with MIC, we were looking at simply we were looking at OD values. And after treating them with penicillin for 24 hours. And then we were looking at the ratio. So if the ratio were, if the ratio was considered 2 or below, we would consider them susceptible. But if they were higher than 2, then we would consider them between 2 to 4, we would consider them paradoxical, neither here nor there. And then if it's higher than 4, we would certainly consider them tolerant. That was our definition. And if you see A909, which is this strain here, you can see that their ratios are 1. So they are definitely susceptible. Whereas O90R, their ratios are 32. So they are clearly tolerant. So those are the 2 strains that we also looked at NEM316, which kind of was a little wobbly. You can see it was sometimes 4, sometimes 16, sometimes 8. So we called it intermediate. That's why we called it a little bit paradoxical, intermediate. But this was firmly penicillin tolerant. This was firmly penicillin susceptible. This too was definitely established many, many times. Moving on. The other thing that we did to characterize it beyond the ratio was, I don't know if you can see this any better. Okay, so in the first experiment, we did a penicillin killing and regrowth tolerance assay, which is basically where we grew them in culture to exponential phase. Then we added penicillin to it. Then we grew it, and then we regrew them in a culture where we added penase to it to basically break down the penicillin, to kind of mimic the situation where penicillin is withdrawn by the physician. So, and then we would wait to see if the bugs grew again, if the GBS grew back again, and we would measure how much it grew. And you can see, maybe I should just go ahead and, can everybody see this? Can you see my screen?
Yes,
Can
we
everyone?
can see, yes, we can
Okay.
see your slide.
Thank you. So if you can see this, then A909, which is the susceptible strain, as you can see, it keeps being low, and O90R, which is the tolerant strain, it's constantly high at all the we looked at different concentrations of penicillin, and at all the concentrations, even at the higher concentrations, it's constantly showing high OD values, which means that they are quite viable. Similarly, we measured cell viability measuring ATP concentrations. We looked at, we extracted ATP initially before adding penicillin, and then we also looked at ATP concentrations after penicillin to look at cell viability. We looked at the ratio to get an idea of viability. And we found that O90R had higher viability, but then we also looked at penicillin-induced lysis, how much lysis occurred, and this was again we looked at this was done by measuring simply absorbance, and you can see that the absorbance went down after penicillin treatment in A909, whereas O90R was not affected. Which means that it wasn't lysed as much. So clearly penicillin cannot break down O90R, whereas it can break down A909. That part we were able to characterize again and again without any doubt. Then we started looking at the sequencing. So we compared by blast, aligning them, their sequences, available sequences of different gram-positive and gram-negative species. We looked at the PBP4 sequences of different strains, and we noticed something that was very, very interesting. We realized that we found a common factor in many of the strains. We realized that the susceptible strains had a specific amino acid. We found that the GD sequence was found in most of the susceptible strains, whereas the tolerant strains had the VI version. We found that polymorphism, we found that pattern. And the part that is not shown here is that we also had access to clinical strains, and about 51 clinical strains. We do not have the IRB yet, which is why I cannot show it here. So we also saw those, we also did sequencing on those clinical strains, and we already, from the clinical strains, we already know which of them are tolerant and which of them are susceptible from the patient's medical files. So the ones that did not respond to penicillin treatment, almost all of them showed that same VI polymorphism. So we knew that we were looking at something that was different. So we found the same pattern over and over again. So when we put it on the at the protein sequence, BLAST2 sequence on the web, we realized that it causes a change in the active site. And that could actually cause a change in the way possibly penicillin binds to PBP4. So when we look at the sequences of A909, you see that at those two specific points, the GV, it has in one, if you go back to the slide, at this point you have G, and then the other point you have at 168 you have G, and 289 you have V. Whereas in the tolerant strain, you have the D and the I. You have that combination, which is something you find even in the other tolerant strains. Whereas if you look at the two susceptible strains, you will find the GV combination. So, and as I told you, we already looked at other clinical strains. The ones that I'm showing you, these are all lab strains, but even in the clinical strains, we found a very similar pattern, and they matched with the clinical characteristic of the strain too. So what we did was we thought that we needed to look at knockouts. So we simply did homologous recombinations. We looked at the parent strains, and then we simply did a swap. We did the GV swap, and we took the susceptible strain and we swapped it with a DI. And we made sure that we did a control to make sure that we were not seeing the results because of our genetic manipulations. So we did the parent strain, we had a GV swap and a DI swap. And then we had the parent O90R, which is a tolerant strain. And then we swapped it with a GV. And we realized that when we swap it, what we were expecting is we should see that the susceptible strain should indeed then become tolerant, and if this is the right polymorphism, then it should be, if this is the right mutation that we are looking at, then swapping it would change the susceptible strain to tolerant and tolerant to susceptible. We should see that if it indeed is the right SNP, the right single nucleotide polymorphism, if it's the right biomarker. And so what we did here is propydium iodide is a fluorescent marker that is non-membrane permeable. It only permeates the membrane if the membrane is compromised, if the cell is leaky, the membrane integrity is destroyed. So you should see the red bars go up only if the cell is affected. So as you can see, the A909 parent after penicillin treatment goes down and O90R is short. This is how it should be, because the cell is not really affected a lot by penicillin. But after you swap it with a GV, you can see that it goes up, which means that now it has become more susceptible. But with A909, which was susceptible, when you swap it with a DI, which normally the O90R has, you can see that its susceptibility has gone down. So we saw what we were expecting to see. But just the opposite is seen in RSG. RSG does just the opposite. RSG measures cell viability. It's redox sensor green, which basically measures the redox level, how healthy is the cell, and it is membrane permeable. So if what it does is that the levels should be low if the cell is not healthy, and the levels should be high if the cell is healthy after penicillin treatment. So because after penicillin treatment, the susceptible cell is not viable, the levels of RSG is low, and O90R is still healthy, the RSG levels are high. But after the swap, A909 has become high now, because now it's not as much affected as penicillin. But O99, after you swap it with GV, which normally A909 has, now it has become susceptible, because now it's not as healthy as it used to be. Now it is becoming affected by penicillin. So that is exactly what we were hoping to see. We also kind of looked at the membrane structure. So we looked at these swapped membranes. We extracted the membrane and then we digested it with mutanolysin. We only extracted the membrane fraction and we digested the peptidoglycan section with mutanolysin, and we ran it on SDS page gels. And as you can see, with A909, the peptidoglycan is not, you can see that it is broken down. That's why you have a smear. O90R, it's more intact. That's why we get a nice band. But with A909GV, it is a smear. O90R DI, it is a nice band. But when you swap it, you can, we would be, with A909 DI, you get the nice band, but O90R, you see the smear. So again, we see the same pattern. We saw somewhat similar results with survival, and we are still at this stage where we are redoing some of this experiment again. But basically, our conclusions are that we show that A909 normally shows a lower rate of survival on treatment with penicillin. O90R shows higher rate of survival with penicillin. And their peptidoglycan, it resists digestion with enzymes, it much better. But so clearly there's something going on with its peptidoglycan. It's stronger somehow, that we know. Now, the penicillin, we also know that it has that particular polymorphism, and when we replace the PVP4D allele is replaced with the G allele, and the V is replaced with the I, these characteristics change. The susceptible becomes more tolerant, and if you swap the other way around, the tolerant becomes more susceptible. So we know that these have a role to play in that phenotype. That part we know, that this play a function in penicillin tolerance. How? We do not know yet, but we do know that somehow this plays a role in the ability of the cell wall to prevent the cleavage. Possibly because of a better cross-linking within the peptidoglycan layer. That's what we think. It's able to play a function in penicillin tolerance, possibly because it plays a role in a stronger cross-linking. That's what we think. Do we know how? No, we don't know yet. So how we want to design the assay? We want to the way we think is, once we know that this is indeed the right sequence, we will use that as a marker. We will use a beacon designer assay. So we will create probes, and if one is for the if it does have the if this is the hypothetical sequence, if it does have the if it does have the tolerant strain, then we can we would have a beacon for it. We would tag it with say, in this, let's just for the argument, let's say we will tag it with a red one, and for the other one, if it's a susceptible one, if it's a regular non-tolerant normal strain GBS, we could tag it with a green one. And then if it encounters a regular strain, then only the probe with the that is tagged with the green fluorescence will bind. This one wouldn't bind. Therefore, you would see this green tolerance. However, you would see a green signal. However, if you have if the sample has a sequence that is tolerant, then it will only the red tagged red tagged probe will bind, the green tagged probe will not bind based on the sequence. And so we will see a red signal, and we would be, so right at the time when the sample, when the pregnant woman is being screened, they will not only be able to say that this is the woman has a GBS infection, they will also be able to tell the doctor that this is a penicillin tolerant infection. So the doctor would know from the beginning not to try treating it with penicillin. So they will save precious time by not wasting time treating it with penicillin. They will move on to other treatments like clindamycin or vancomycin. That was my presentation. Thank you so much.
Thank you very much, Dr. Paramita. It was very nice presentation, and we hope all the audience have benefit with this presentation. Okay. Any audiences having any questions and query regarding the presentation? Anyone having one query? I think in Indian scenario, if we see in case of this this GBS prevalence as compared to the Western population, I feel in India it is lesser. What do you feel so, ma'am?
Yeah, actually, I felt the same thing, because I think in here actually GBS is a big problem. I think in India, neonatal mortality is much more because of nutritional fat, because of nutrition and health reasons, opposed to something like GBS. I actually completely agree with you. Something I see all the time. I feel, yeah.
Yeah,
I
because
teach, I teach infectious diseases. Believe me, when I try to teach malaria or TB, these are really not something that are a problem here at all. This is something I face all the time.
yeah, definitely. Because what I see, there are few studies in ICMR, they also claim that there are incidences of premature deaths or premature, you can say, delivery because of GBS. But still we can see the incidences are very low. And one of the reason maybe I feel the reporting is less in India. That is, of course, one thing I feel. Because no one is,
True.
yeah,
But also I feel like it's probably just also the numbers.
yeah.
I feel like the total number, if you look at, importance and resources are always allocated by numbers. And I feel like the numbers of neonatal deaths are so much more because of other causes. Probably those few deaths because of GBS is very, very minuscule. So it's probably not considered important. Here, if you see, it's really the number one cause of death. I mean, malnutrition is never a problem. It's really not something, it's unheard of. I know it, but yeah, it's still something that we are dealing with in India. So, because I still attend a lot of the UN lectures. We are part of the UN Academic Impact. So I'm certainly familiar with the top 10 commandments of the UN. So this is something that's very close to my heart. I work very closely with women and children issues all over the world, not only in India. So, and I come once in a year, I will come to India for medical missions and service. So we see this a lot. So yes, this is something, if just to let you know, in any case, I'm always here once a year, usually in December. Usually we come with our students for provide, if any of you are involved in any of these projects, please let me know. I'm always happy to volunteer.
Yeah, sure. We have a medical college here, and we all have. I'm director of the family program here as well, and then maybe we can find some project, and then we can collaborate in future with you as well.
Absolutely. Our
Thank you.
students are always happy to come and do all of these. They're always very excited to come and work. And thank you so much for giving me the opportunity. I wish my students could be here too, but thank you so much for the opportunity.
Yeah, ma'am, Paramita ma'am. This is Dr. Somnath, actually one of the chairperson in today's session. Am I audible? No? Am I audible?
Yeah, we can hear you. Please, Somnath, Dr. Somnath.
Yeah. So, ma'am, first of all, I would like to congratulate you for your nice presentation and all. And moreover, though this conference in commemoration of International Women's Day, so belated Happy Women's Day to you. And,
thank you so much.
And all participants, those who are participated in today's questions. It's my last question is, am I audible? Hello. Hello. Are you audible?
Yes, Dr. Somnath, you're audible.
Yeah. Hello, ma'am.
Yes,
So my
I can hear
question
you.
is, Yeah. It's okay, ma'am?
Yes.
Yeah. So my question is, the penicillin doses interval should be reduced or can be reduced?
Penicillin doses. That's actually a pretty good question. Here, actually, the guideline is to not use doses more than necessary at all. We try to we start from the bare minimum. That's what the guidelines suggest. There's a lot of antimicrobial stewardship. We teach that here. Because a lot of my research is on antimicrobial resistance. I just finished writing in 2021. I just released 4 chapters on emerging community, and this is something we update every 4 years. So I know that there's so much like emerging hospital as well as community acquired antimicrobial resistance. So I can't help but say that the bare minimum dose is probably the best thing to start with, even for penicillin. But not only for
antibiotics, for all the antibiotics.
All antibiotics, yes. But in India particularly, yes, this is a problem because yes.
So, and one more question is like, can you build a tolerance to the penicillin?
I believe so. Penicillin tolerance has already been reported in, say, as I told you, it has been reported in Staph aureus and Streptococcus in group A strep. Group B strep, there has been reports, but really nobody came up with a mechanism. But there are reports. Problem is that nobody has been able to come up with a proper mechanism yet, but there are many clinical reports. There's a very fine line between resistance and tolerance. That's the problem. I think that's why it's very important to characterize it. I think some people characterize it. I think that's where the scientists differ. There's still a debate between how you're going to call it a resistance or a tolerance. To me, the definition of resistance should be that it will keep on growing.
Okay.
And tolerance would be that it will stop, because it cannot be detected. If it doesn't grow, if its numbers become so low that it cannot be detected by PCR. So the doctor is thinking that it is cleared. But the moment you withdraw it, it starts growing. That means the cells did not die. So if it's capable of growing back, that means the cell were viable. Even if 2-3 cells are viable, it can grow back. So then how then it's tolerant? Resistant means that it should still keep on growing. You should still be able to detect it.
Exactly.
That is how I see it. But there are groups of scientists that there is some debate. But really, that is the only debate. But yes, there are lots of cases that there is reports.
Thank you so much, ma'am. Thank you so much for your.
Yeah, very good discussion, Dr. Paramita. So, as you said that it's a resistance, is an ineffectiveness. So, when the drug is not effective on the microorganisms, then only it will develop the resistance. So clinically, what we do, so when the patients, experience of the resistance, may chance to develop some drugs, particularly with the Escherichia coli or H. pylori infections and all. These infections with the one antimicrobial drugs is very difficult to eradicate or to inhibit the growth of microorganisms. So that we used to give the combination therapy to avoid the resistance. So the combination therapy is very important when we treat the patient based on the bacteria. So based on the bacteria only, we go for the treatment, the pharmacology aspects, the clinical treatment after the diagnosis. So based on the bacteria only, we used to go for the treatment. So this is a very interesting topic. to
That is
learn.
exactly why I feel like at the time of screening, if we are able to look at the tolerance profile or resistance profile, then we are not wasting time, by treating first with the wrong antibiotic. And then, because when a baby dies within 5 days, it's very important. There's no
In our
time to waste.
country, especially in India, so the issue is inappropriate the prescription of antibiotics is one of the the major threat to our society. So what we have to do as a pharmacist, before dispensing of any medications, we have to read through the prescriptions or any drugs. What purpose we are dispensing of the drugs, is there really is a necessary for the patients, then only we can dispensing of any antibiotics. But in abroad, in other countries and all, without any prescription, they won't give any antibiotics. But in our country, without prescription also, we can easily buy the medication. This is one of the drawback of the society. The OTC medications is a very is one of the issue in India. OTC is a normal non-steroidal drugs. Okay, fine. But when the patients are taking of antibiotics, really they do not know what purpose the antibiotic they're taking. Some of them, if they common cold also, they used to take some fluoroquinolones or any macrolides. Is really?
Of course, I grew up in India. I grew up in India. I'm also guilty of that. But I have learned over the years here.
So from there.
Yeah, but here in pharmacy practice,
I think president also knows,
right. I mean, here we grew up. Somebody has a cold, take amoxicillin.
But it's okay. In the future, everything will be changed. We hope that everything will be changed through our association. The strong.
Yes, that's the idea.
Give it to
but also
the health department. Maybe in the future, think on it. We'll see. Our
I don't know if in India, do they have the antimicrobial stewardship programs? In the US, in pharmacy schools, the pharmacy practice programs, they have an antimicrobial stewardship program, which I'm hoping that they would also introduce in pharmacy schools in India.
In India, it is not there, unfortunately, these days. But I'm hoping, in future, maybe we can recommend this.
Yeah. That's true. Thank you, Mittal sir. Thank you very much.
Thank you so much for the discussion.
Paramita, it's a wonderful session.
And
thank you.
Dr. Mittal, Dr. Vetri, and Dr. Somnath for the wonderful discussion. It will definitely benefit our pharmacy professional in their research project.
And thank you so much, Dr. Dahia, for the opportunity too. Thank you so much for APP for giving me the opportunity.
Yeah. So with this, without taking not much time, I will invite Professor Sundar to proceed for reading the profile of next speaker, Dr. Sivkanya.
Thank you, sir. So it is my great privilege to introduce our today's second speaker, Dr. Sivkanya Floria. She completed her B Pharmacy from Amaravati University, India, M Pharmacy from Department of Pharmaceutical Sciences, Nagpur University, India, PhD Pharmacy from Amaravati University. She has 17 years of experience in teaching and research. She is presently working as an associate professor in pharmaceutical chemistry, AIMS University, Malaysia. She has published different massive open online courses such as the basics of H1NMR and data interpretations at udemy.com, principle and practice of production management and introduction to glycosides and resins at openlearning.com. She has published 4 books in the title of Fundamentals of Pharmacoeconomics, Principle and Practice of Traditional, Complementary and Alternative Medicine, Principle and Practice of Industrial Management, Spectroscopy, Fundamentals and Data Interpretation. She has filed 1 patent in the topic of composition for the treatment of urolithiasis. She is a recipient of 4 FRGS Grant Malaysia in the total amount of 5,55,600 Malaysian ringgits. that is approximately equivalent to 1 crore in our Indian rupee. She has published 72 research papers and presented research articles in 65 conferences. She is guiding BPharm, MPharm students and 6 PhD scholars also. She is a life member of various national professional bodies like member of Indian Society of Analytical Scientists, Indian Pharmacy Graduate Association, Delhi Pharmacy Council Welfare Trust, Indian Pharmaceutical Association, Association of Pharmaceutical Teachers of India. She has different achievements like gold medal in DPharm, qualified in GPAT with 83.76%, received appreciation award from AIMS University, Malaysia, received best presentation award at 19th International Conference on Medicinal Chemistry and Molecular Pharmacology in Singapore. So this is the brief introduction about Dr. Sivakanya Floria. I hand over the session to Madam. Please, Madam.
Thank you, Sundar sir, for a nice introduction. Thank you very much. First of all, I would like to thanks to all the APP members and especially Dr. Daya sir and Dr. Sunita Daya madam for giving me an opportunity to share my. This is just a platform where we can share the knowledge with you all. So this is just a sharing of knowledge, and I would like to thanks Dr. K. Padmalata, Principal of Vijaya Institute of Pharmaceutical Science, and the management who is supporting to organize this conference, 2 days conference. So here I'd like to share my slides. This is just a sharing of knowledge. I'm not expert for this particular topic, but still I'm just sharing knowledge for this particular topic. Okay, so today's talk of my presentation is endophytes, the research paradigm. So as you can see, the endophytes actually is the word derived from the endon and the phyton, which means the microorganisms which are within the plant cells. We call them as endophytes, and basically there are 300,000 plant species found in a world, and in every plant at least one endocyte reside inside the plant cells. And if any plant don't have any kind of endophytes, that means it is more vulnerable to the environmental stress, pathogenic attacks like what I can say, the abiotic and biotic stress, that if endophytes are not there. So basically, what are endophytes? Just now I told endophytes reside inside the cells. So you can see this is a picture where we can see this is a scanning electron microscope picture where we can see the endocytes inside the cells. Then the endophytes actually basically the scientist who discovered this particular name is D. Berry in 1866. He defines the endophyte as any organism that grows within the plant tissues are termed as endophytes. So basically endophytes are a group of symbiotic group of microorganisms, and they may be bacteria, they may be fungi, and they colonize inside the different locations of the plant that I will share later with you. So basically, if you can see, just now we have seen the bacterial endophytes. This is the picture showing the fungal endophytes. You can see here on screen. Then the microbes in plant. There are different types of microbes in plant. One is we call them as epiphytic microbes. Second, we can call them as rhizophoric microbes, and third, we can call them as endophytic microbes. So basically, epiphytic microbes, they are you can observe them. They can found in the aerial part of the plant, especially leaves. We can say the flowers. We can say. Then rhizophoric microbes, basically they are found in the nodal system or root system of the plant. Basically, internal nodes. Maybe if you have learned during our maybe the 12th or 10th standards, that time maybe we have learned these all like kind of the systems where especially for nitrogen fixation, these microbes are very much important for the growth of the plant. And the last one, which is we are interested, that is endophytic microbes. These are the microbes which are present in the internal tissue, in the internal cells of the this aerial part as well as root tissues, and them we referred as endophytes. So basically 3 types of microbes as we can see on screen: epiphytic microbes, rhizosporic microbes, and endophytic microbes. So if you can see on screen, I have showed here picture how they look like. So you can see here endophytic microbes are there, epiphytic microbes are there, and rhizosporic microbes are there. So basically we have seen the difference between endophytic microbes, epiphytic microbes, and rhizosporic microbes. So life cycle of endophytes. Basically endophytes, as we can see, any plant, the growth of plant starts from the seed. Okay, so you can see basically the endophytes are found inside the seeds, and when plant grows, it germinates, and after that there will be the root system. So most of the endophytes in third stage, they will concentrate inside the root system, and after that, once plants start to grow, you will observe in every part of the plant, the endophytes will spread inside the cells of the plants. So what are the different locations where we can found endophytes? So we can get endophytes in stems. We can found endophytes inside the roots, petioles, leaf segments, fruit, buds, seeds, and flowers. So most of the plant parts are having these endophytes. Even I would say this is not just uh, having inside the plants, they are available inside the marine resources also, but that I have not covered here. So what are the benefits of endophytes? So they are helping plant to tolerate to the heat, the temperature variation. The plant can sustain in any kind of the temperature climate change. Then they help to go for the nitrogen fixation in order to plant for the plant growth, and helps to increase the resistance toward the pathogens. That is one of the benefit of the endophytes, and they are also helpful to remove the contaminants which can solubilize, especially as you know, these endophytes act as a phytoremediation. Later I will discuss how they are useful as a phytoremediation. That part I will discuss lastly. Then they are helpful for the protection against the biotic and abiotic stress. Abiotic means like temperature variation, climate variation, excessive rainfall, excessive temperature. Then biotic means some like a microbial attack is there. So there these endophytes will act as a shield for the plant, and they support plant, they protect plant, and to facilitate the nutrient absorption. This is one of the important aspect of the endophytes. And in case of the synthesis, biosynthesis of various growth hormones like auxins, ghrelins, all these are synthesized with the help of these endophytes, and they are helpful to increase the plant growth hormones, especially auxins. Just now I discussed cytokinins, ghrelins. These all are required for the growth of the plant. So you can see here, this is the picture. All images are not mine. This is just a sharing of knowledge. So whatever image I have taken, that is acknowledged, I'm acknowledging. And this is the picture where you can see these endophytes are like how these are working for plant protection. So when any kind of the pathogens of herbivore attack on the plant, then the plant will send a signal to the for production of chemicals, and these chemicals are produced with the help of endophytes. So you can see on screen. So the way these endophytes are worked. Then what are the different bioactive compounds derived from the endophytes? So you can see most of the, you know already about the secondary metabolites like alkaloids, flavonoids, phenolic acids, quinones, steroids, saponins, tannins, terpenoids, and xanthan. So most of the secondary metabolites are derived with the help of these endophytes, and you can see here examples. These are the uh, endophytes derived chemicals which are used as antibiotics, antifungal drugs, immunosuppressant drug, antiviral drug. As you know, the current era, most of the research is shifting towards the natural resources, and most of the researcher throughout the world, I think, working on this particular segment. So one of the area which we can focus, we can go for searching of the endophytes with the novel medicinal moieties. So there are mechanisms by which these endophytes can help for the plant growth. So you can see two ways are there, one with the direct mechanism and another is indirect mechanism. So you can see these endophytes, as already discussed, helpful for the nitrogen fixation, phosphate solubilization, and they're also helpful for the releasing the growth hormones. Then there are two types of endophytes. One is systemic, true and non-systemic transient endophytes, and another one is claviciptious and non-claviciptious endophytes. So you can see there the systemic and non-systemic endophytes. These are basically microorganisms which can reside inside the tissues with their whole life cycle, and they participate in a symbiotic way. Means they won't harm the plant. They will helpful for the growth of plant and without causing any kind of disease. You might be knowing there are bad bacteria and good bacteria or fungi, but so here they will act as a good like a microbes which can help for the helpful for the growth of the plant. So the concentration and diversity of endophytes will not change uh, with the variation of the environmental condition. Just now I told environmental condition may be the change of temperature, the change of the rainfall, maybe the excessive heat, or that can be. But these kind of the endophytes won't change, the concentration won't be affected with the environmental changes. The next classification we saw, we can see here non-systemic or transient endophytes. So these microorganisms actually basically pathogenic when the environment changes. When there is a change in temperature or change in climate or too cold climate, then they will affect on the growth of plant and they will cause the disease to the plant. So they are called non-systemic or transient endophytes. And then there are other classification, claviciptious endophytes and non-claviciptious endophytes. So we can categorize them in a class 1 claviciptious endophytes and non-claviciptious endophytes, class 2, class 3 and class 4. So what is claviciptious endophytes? Actually, claviciptious endophytes basically categorized under the class 1 endophytes. They basically make their colony inside the shoot of the plant, and they are transmitted inside the what I can say, the from root to the top, from the aerial part they will transmitted from host to host by vertical transmission. So this is specially you can see in case of the fungi. So you can see this is the fungal endophytes which can transmitted through from root system to the aerial part of the system. Then non-clavicipitous endophytes, again, we just now seen the classification, that is class 2, class 3 and class 4. Basically, these are available everywhere in the plant, and these are actually helpful for the growth of the plant. And this is actually what I can say, the polyphyletic group of organism is the diversion of the group of microorganism you can find inside the tissues of the plant. The next part is a collection of sample. Now it's coming on to the research. We just now discuss what are the endophytes, how the classification goes on and where they are located. Now how to go for the research using the endophytes. So the first thing we need to do the collection of sample. So once we collect the sample, we need to make sure whatever sample we are collecting, that should be healthy, that should be free from disease, should be whatever free from the disease and symptoms, so that whatever the endophytes we are isolating, that are pure endophytes, not the pathogens, not the microbes which can causes the diseases. So we don't want that. We are interested in endophytes. Then after collecting the sample, you can process the sample within a 24-hour, and once you collect the sample, you can keep at 4 degrees Celsius in a plastic bag, or the purpose to keep at 4 degrees Celsius, so that we can keep the tissues of the plant fresh, the cells should be fresh, and we can avoid the microbial drain. The endophytes should not, because ultimately these are the microbes, so we don't want to lose them. So for that purpose, we need to keep at 4 degrees Celsius. So we can use different part of the plant. We can use leaves, we can use stem, we can use root system. As I mentioned, there are different locations depending upon the plant which you are working on. By selecting the plant, you need to select the correct part of the plant, and then you can go for the research for the endophytes. The next is you can use, as I discussed, you can use various part of the plant, and as these endophytes are present in everywhere, but most of the as we can see from the life cycle, most of the these endophytes are concentrated in the root system. So density of the root system inside the root system is more. Then once you collected this endophytes, you need to go for the surface sterilization and fragmentation of the plant tissues, and the strategies for or methodology or procedure will be same, except you need to make sure for fungi you need to use the different medium. The antibiotic may be different, the incubation period may be different, the molecular marker, if you're using that, are also differently you have to use when you go for the fungi and bacterial endophytes. So you need to make sure you are selecting the proper growth medium. Then once this done, now the surface sterilization. Once you collected the plant, you make sure that the plant is healthy. You make sure that it is free from the any disease. Then after that, you need to go for the surface sterilization. So what is the purpose of surface sterilization here? To isolate the microorganism that is of our interest, that is endophytes. We don't want epiphytes. Just now I have discussed different types of microbes. So one is epiphytes, one is endophytes and rhizosporic microbes. So we are not interested in epiphytes, we are interested in endophytes. So for that we need to go for the surface sterilization. So how to go for surface sterilization? You need to rinse with the sterile water 5 to 10 times, and after that you need to wash with the ethanol, 70 to 75% ethanol. And once you have washed with the ethanol, then you need to rinse again with the distilled water. And once this done, then you have to this is the picture which showing how you can go for the surface sterilization. And then once you have done with the uh, uh, sterilization with the water, then after that you can use, if you want to use the other chemicals also, you can go for the sodium hypochlorite, you can use sodium thiosulfate. And after once you done with the sterilization, you can use this water as a negative control. Once you use you went with the sterilization, then you have to confirm whether the material or the plant material, the specimen which you have collected is actually is a free of microorganism or not. That we can confirm by inoculating with the suitable medium. As I told, if you are going for the fungi, you need to use a different medium. If you are inoculating for the bacteria, if you are looking for the bacteria, then bacterial endophytes, then you need to use the another growth medium. So once this done, you need to make sure your plate is clear. If it is clear, then that confirms there is no epiphytes grown on that particular part, or there is no epiphytes. So that confirms the complete sterilization of the plant. Then once you have done with the sterilization, now the time to isolate and identify. So how to identify this? You can go with this small pieces of the this. You can just make the pieces of this particular sample which you are looking for the endophytes, and you can go for the inoculation and incubation. And finally, you once you done with incubation, and you can see the growth of the pure colonies of the endophytes. So this is which we are interested or the purpose of particular that research, to find out the endophytes, whether it's a bacterial or fungi. So we want the pure colonies of these endophytes. So once we got the pure colonies of endophytes, this is the method which I just now shown you. Collection of plant, then after that surface sterilization, fragmentation, inoculation, incubation, and pure isolate of endophytes. So once you have done the endophytes, now it's time to find out what kind of the endophytes you have isolated. So there are two methods which you can use, that is based upon the molecular base identification. You can extract the DNA by using the cetyl trimethyl ammonium bromide method, or you can use the DNA kits to know the molecular structure of this particular endophytes isolated. So how to go for the molecular structure? So you can use the kit, DNA kit, or the method which I mentioned right now. Then once you have done the identification, you need to go for the amplification with the suitable marker. And I think previous speaker already, as she's working on this penicillin resistance, so I think she particularly worked on this particular. She has given idea about the molecular base identification. So with the help of PCR machines, you can go for the amplification, and once you have got the amplicons, you can go for the electrophoresis. And once you have done the electrophoresis, you can go for the DNA sequencing. And once you have done with this DNA sequencing, this is the flow where we are just isolating the endophytes, and you can see here there are two types of endophytes, bacteria and fungi endophytes. And once you have done the isolation, you need to go for the morphological characteristics. And how to go for the morphological characteristics? So you need to go for the just now the previous speaker mentioned about the blast, using the blast sequencing. So you have to go for the blast sequencing, and once this done, then you can confirm the what kind of the endophytes you got, like bacterial endophytes or fungi endophytes. The next, coming on to the application of endophytes, so you can endophytes have numerous kind of the applications. First application, like agriculture, so they are helpful to increase the crop yield. And here one of the example you can see, Formiforsa indica, which is fungus, and which is helpful uh, for making the symbiotic relationship between the other plants. So this is one of the important aspect of the these endophytes. And these endophytes, you can see various applications. Just now in during endophyte application, I told how they are helpful. They are helpful in terms of biofertilizers, because right now we are also looking for the new instead of using the chemical fertilizers, we are looking for the biofertilizers which can help, which can reduce the consumption of chemicals, and towards more towards the bio materials. So we can use this one of the endophytes as a one of the important tool in agriculture segment. Then another application in form of the biofuel we can use. So these endophytes can able to make the terpenes and pollenes, and these terpene pollenes can be used as a biofuels. And here one of the example you can see, endophytic fungi Nagrograna macnoni, which is isolated from Gonza ulmifolia, which from that particular plant, pollen isolated, which can be used as a which has a property like a gasoline, so which can use as in terms of a gas. So you can see here a picture. There are different applications of the biofuels in terms of we can see different the biodiesel plants we can use. Then as I told, these endophytes are helpful as a phytoremediation. So phytoremediation means it will helpful to break the pollutants. So how they are helpful to break the pollutants? Like they will either chelate that particular the pollutants, the chemical, or they will stimulate the microorganism in a soil to break down the organic pollutants. So this way they are helpful in case of the phytoremediation. So here you can see this particular endophyte example here, fungi Pestilopsis microspora, which is helpful to digest polyurethane plastic as one of the important one of the important we can say the pollution currently we all are facing. Like a polythene is one of the biggest threat to the world everywhere, especially I can say in our this India we have this kind of the pollution at high level. So for that purpose we need something which can help us to reduce this the pollution, especially the plastic pollution which is causing. So these endophytes are helpful for the phytoremediation, and you can see here how they can helpful in making this as a for chelating the pollutants. You can see uh, the process. Then lastly, which is as a pharmacist we are more interested, In case of the drug discovery, these endophytes are very much important, and these endophytes actually, as I shown, these are responsible for the producing the secondary metabolites, and these secondary metabolites can be useful as a lead molecule for in case of the drug discovery. And you can see here these can be the good resources for the antimicrobial, arthritic drug, anti-diabetic drug. And one of the example you can see here, one of the example here is a Taxol, which is very famous drug, which is produced from the fungi, endophytic fungi, that is Taxomyces and arini. It is isolated from the yew plant that is called Taxus brevifolia, and this particular plant is responsible for production of Taxol, which is used in treatment of the cancer. So you can see this is the market product. This is just information purpose. So you can see there are numerous kind of the endophytes and their activities available. You can see the penicillium just now. The previous speaker talked about the penicillium. So one of the resources can be the endophytes, where from this penicillin is derived. And you can see number of examples are here. So you can see these are helpful also as a cardiotonic drug. The bioactive components isolated from this particular cladosporium, which is which is helpful as a cardiotonic drug. So these are the numerous example you can see here. And these are the few papers which are focusing on the research part of this using the endophytes. You can see evaluating antibacterial, anti-cancer activity of crude extract of bacterial endophytes, then anti-cancer activity of bacterial fungi, endophytic fungi, you can see here. So in nutshell, I would like to say endophytes are the microbial species that resides inside the plant tissues symbiotically, and they produce bioactive compounds, and these are bioactive compounds which are responsible for the growth of the plant, for the production of the for the protection of the plant. And these are not only helpful for the plant, these are also helpful for the human beings. Like they are we can use them as an antibiotic, antiviral drug, we can use in food industry, we can use as a to stop the pollution, as a biodiesel we can use them. And finally these endophytes, as just now we discussed, important in they have a important role in case of biodegradation. So overall, the endophytes are the important part of the natural environment. So these are the references you can see, and I would like to thanks to APP organization for giving me an opportunity. And this is one of the best society which is giving opportunity to share knowledge and to learn something new every time. Whenever I attend any kind of APP conference, every time I will come to know the new things happening in the world. So thank you so much APP, and thank you so much again Vijay Institute of Pharmaceutical Science. Thank you so much. So
Thank you, Dr. Florio. Over to the judges.
Yeah, very nice presentation. Nothing means no doubt about it. Okay, so I'm having one doubt about on your presentation, madam. Am I audible now?
yes, sir.
Am I
You are audible.
Actually, I want to check the secondary metabolites of the endophytes. Okay, whether the endophytes have the similar phytochemicals or not. Okay, so and also I have lots of endophytes isolated from the medicinal plant. So can you suggest me to what should I do?
If you are done the isolation, it's good. Then maybe you can go for the characterization of that particular endophyte isolated compounds, bioactive compounds. You can
Okay.
go for the characterization so that you can confirm the structure of that particular compound, whether it's an alkaloid or I think you are aware about the characterization procedures. Okay, the different methods are available, so you can go with that. Uh,
So, madam, which is the best stage for the isolating of endophytes?
I think it depends on the plant and the place and the environment. So it's not that a particular like a particular, as I shown in my presentation, there are different locations, and the concentration of the endophytes depends on the climate and environment also. So it's not necessary. Miss, I can comment which part you should focus.
Okay. Thank you so much, ma'am. Over to Piyush Mittal, sir.
Thank you, sir. Thank you very much for a very comprehensive presentation on the endophytes. Actually, I am from the clinical pharmacy, so this is a new topic for me to learn about this one. And my area is just restricted to the hospitals and the wards and experiential learning of the student. So this is a quite new topic for me. And then thanks for enlightening me on this topic as well. And we know that there are new drugs in pipeline, and then we have a microbial resistance as well. And then surely, if we are having a cure of disease with the help of these kind of the natural products, okay, it may be helpful for the society. Thanks a lot. Okay.
Thank you.
Thank you, Dr. Floria, and the judges for the nice discussion. And session was quite knowledgeable. And students can utilize the facts discussed in their research projects, and it will be of significant value. So thank you both the speakers and the judges and other guests who attended this session and interacted during the session. So this completes our scientific lecture session. And now what is time in India?
12. Sir.
Hello. So it is 12. So we are on time. So it is, as per the schedule, it is lunch break, and then we will proceed at 1:10 for the oral competition, where 8 presentations will be made today by different research scholars of different states of India. And I think Prof. Mittal, you will continue as judge of the session.
Sure, sir.
Oral session, and other judge will join during that time. So I thank you all for so nice lectures and so wonderful discussion, and it will be useful for us.
Hello.
Thank you.
Thank you so much, sir. Thank you so much.
Thank you, sir. Thank you, Prof. Rajiv Dia.
So hope to see all of you at 1:10.
Thank you.
I think somebody has raised their hand, sir, regarding. I think Dr. P. Bharat.
You can carry out the discussion. So, thank you.
Hello. Sir, I am audible to you?
Yes, Dr.
Yes, sir. Yes.
Yes, sir. Myself, Dr. Bharat, HOD, Pharmaceutical Chemistry Department from Anwar Ulum College of Pharmacy. Hello.
You are audible, sir. You can continue, sir. No problem.
Ma'am, it was a very nice presentation, ma'am. You have such a nicely elaborated. A very informed student also can start research on whatever the topic we explained today. I have a few questions, ma'am, 2-3 questions.
Sure.
So can you throw some light on what are molecular markers we can use and what are amplicons?
Sir, actually, this is more towards the biotechnology part, and this work actually particularly is done with the help of biotechnologists. So we have a department where we normally take the help of the biotechnologists. Sir. So I'm not sure about the markers and what kind of the things we should use. Okay, maybe if you give time or personal also I can inform.
But Dr. Basu is the biotechnologist. If she can address something related to that.
Can you give me a little more detail about your questions? Yeah, I work with biomarkers. Yes.
Yes, ma'am. What are the molecular markers we can utilize in this particular discussion? Ma'am told to isolate the endophyte. We can go for its application part. What are the molecular markers? Means we need to go through the chemicals and requirements for this experiment. So I'm asking about what are these molecular markers can be purchased.
So typically for biochemical for any so for molecular markers, typically when it's a protein, we look at the gene that is responsible for we try to identify the gene that expresses the protein. So the gene that codes for the protein, once we identify it, usually it is done by a blast search. You keep looking for that same gene. You compare the sequence of the gene. BLAST is nothing but an alignment comparison. You align the sequence in different species, related species. In this case, if it's in like in my case, I would look at similar kind of bacteria and look for the same gene in 5 different bacteria and compare the sequence and see if there is any minor difference in the sequence.
Okay.
And once I look at the difference, that helps me to find out the common region that looks alike within the gene. Okay. And that part will become the biomarker. There are if you go into NCBI BLAST, there is a lot of free software that will help you identify the biomarker.
Okay.
There is GenSec. There is a software called GenSec. If you go into NIH has a lot of free software that will, through PubMed, you'll be able to find a lot of free software that will help you identify these biomarkers. You'll have to know what gene you are looking for, of course. But once you put in a search, it will pull up different microorganisms. Say if you're looking for something in, say, gram-positive bacteria, you look for it in 5 gram-positive bacteria, 3, 4, 5 gram-positive bacteria. It will pull up the similarities in the sequence, and it will identify the region that is exactly identical. It will pull up a region that is, say, I don't know, 24, 25 base pair long. Okay. And that will become your biomarker. There are some criteria. You want an area where there is no hairpin loop, where there is no mismatch, You want an area which will bind well with a probe. When you design a probe, you need a primer. For these kind of biomarkers to design an assay, you will need, usually biomarkers are made for PCR-based assays, and so you need another piece to hybridize with it. So you need to design it in a way that it will bind without any mismatch. Complementarily, it will bind, DNA binds complementarily, right? So there should be no mismatch.
Okay, madam. Thank you.
But there are softwares, there's free softwares that you'll get.
Madam, one more question. What are the basic equipments required, madam, to carry out this?
You need an RT-PCR machine for this kind of research. For biomarker identification, you need nothing. You need a computer in silico. But for RT-PCR, you need a qRT-PCR machine. We don't even use. Well, you could do it the way Dr. Floriat said. You could do a regular. You could do a regular RT-PCR, which is simply reverse transcriptase, and then amplify it using a PCR. And then you could do it the hard way, like she did. You can run it on gel, and then look at the band. We actually don't do the gel electrophoresis part. We actually do it in real time. We actually do a qRT-PCR, which is a quantitative real-time reverse transcriptase PCR, where instead of running it on gel, we tag the product, the PCR product, has a fluorescence tag added to it. So every time a new copy is made in the PCR amplification, the PCR is nothing but it's like a Xerox copy. It keeps on making new copies. So every time a new copy is made, the fluorescence signal gets increased, and that gets detected by a recording device. So every time the DNA gets doubled, the fluorescence signal gets increased. And that's what the qRT-PCR measures. So it will tell you that there is more copies of the DNA. So you don't necessarily need to run it on the gel to see that you have more darker band, to see that you have more DNA. But but that's also another way to, if you don't have the detection system, then yes, you would run it on gel to see that your band will become darker, to know that you have more DNA. That's definitely another way to do it. It's just more labor intensive. That's it. More time taking. But if you have the quantitative qRT-PCR machine, then it does it together.
Okay, thank you, madam. Thank you very much for clarifying the doubt. I thank Dr. Padmalata madam and other organizing committee members, Piyush Mittal sir, Somnath ji, the APP member. Thank you, sir. Thank you for organizing this beautiful conference. Thank you.
Professor Padmalatha, you want to conclude?
Yes, sir. The program went on very nicely. The talks were very useful. The interactions were very healthy, and the participants raised the questions very enthusiastically. So this type of platforms will help the students and researchers to learn what is happening all around the world in the pharmacy field. So this is one of the best platform, and I'm very happy for associating with your APP and doing the programs. Thank you, Mr. Raju Dahiya.
Thank you, madam, and all speakers and judges and guests. So hope to see you at 1:10 then. Good afternoon.
Thank you so much. And I'm happy always to collaborate if anybody is interested. Bye.
Yes, ma'am. Thank you so much. Thank you. Bye, ma'am.
Bye, Dr. Zahia. Bye, everyone.
Bye, bye. Bye, bye.
ସମାନ ଅଛି ସମାନ ଅଛି ନା ଭାଇ ନା ଦେଖିଥିଲେ ବା ସମାନ ଅଛି ଦିଟା ସାର୍ ସଞ୍ଜୟ ସାର୍ କରିକି ଯାଇଥିଲେ ଆଠ ଯୋଉଟା ହେଇଥିଲା ଖାଲି ମିଶେଇକି ଲେଖିଦେବୁ
ଖାଇବା ଟାଇମ ହେଇଗଲାଣି
ସାର୍ ଭୋକ କଲାଣି
ଲଞ୍ଚ ରହିଲା କଣଟା ସେଟଲମେଣ୍ଟ ହେଇଯାଉ କଣ ସାର୍
ଦେଖିଲେ କାଲି ମନ୍ଦିର ସାମ୍ନାରେ ରହିଲା ଯେ ସବୁବେଳେ କଣ ଯଦି ଯିବ କି କୁଆଡେ ଯଦି କୁଆଡେ ଯିବ କି ତମ ହାତ ଦେଖିଲେ ତ ସାର୍ ଚାବିଦେଲ କହିଲେ ଗାଡି ଚଲେଇବ ଅନ୍ୟ ମନସ୍କ ବୋଧେ କାଳେ କୁଆଡେ ଯିବ
Okay.
Colleagues, good afternoon. Welcome again. So our first session went well with the two lectures of Dr. Basu and Dr. Sivkanya, and now we have the another session, which is a oration competition. It is in memory of late Sri Tarsem Jain, and we have received 8 presentations for oration, so which will be 15-minute each, and it will be followed by 5-minute discussion. We have received the entries from different states, so already I have forwarded the schedule to you with the topic. For this session of oral competition, Dr. Sunita Dahiya, Assistant Professor, Department of Pharmaceutical Sciences, School of Pharmacy, University of Puerto Rico, will be the Scientific Committee Chairman, and followed by 4 chairpersons: Professor Piyush Mittal, Professor of Pharmacy Practice at Tirthankar Mahavir College of Pharmacy, Tirthankar Mahavir University, Moradabad, UP; Dr. Madhu Gupta, Associate Professor, Department of Pharmaceutics at School of Pharmaceutical Sciences, DPSRU, New Delhi; Professor Suresh Chanupati, who is Professor of Pharmaceutical Analysis at Nalanda College of Pharmacy, Charapalli, Nalgonda, Telangana; and Professor Anju Goel, who is Professor of Pharmaceutical Chemistry at BN Institute of Pharmaceutical Sciences, Bhopal Naval University, Udaipur, Rajasthan. So all the chairpersons are needed to evaluate each of the candidate out of 10, and at the completion of 7th candidate, please send your score sheet to at presidentapp11@gmail, so that I can start compiling the score, and last entry immediately after the completion you can send, because we have just 5-minute gap in the award announcement after that. So each participant will take care of their time. No extra minute will be given. It will be 15 minutes. A reminder before after 13 minutes can be given to each participant, so that they can compile up their presentation within 15 minutes, and a 5-minute discussion where we can save time if questions are not more. So with this, I hand over the session to the chairperson or the scientific chairman, Dr. Sunita Dahiya, and she will read out each candidate little biography, like a topic and affiliation and name, and regarding score sheet I already guided. So, and I need to mention that the first person, Veda Pal. So she is present, Miss Veda Pal.
She seems to be here.
Okay.
But Veda, I see her name in the list of participants.
Devani is also
Yes,
there.
sir. Yes,
Okay.
sir. Good morning, sir.
Okay. Then Megha Joshi, she is not appearing. Then Jenny Christian, Uma. Okay, because I asked to be present before 10 minutes, so we can't expect the last candidates. Okay, anyhow, Dr. Dahiya, you can lead the session now. Thank you.
Thank you. Okay, we have our first participant, Ms. S. Vedapal Jayamani. She will be delivering a talk. But first, let me check whether she is here. Ms. Veda, are you here? Ms. Vedapal? And she Ms. Vedapal is,
I think she is muted, madam.
but I see her mic unmute and
You can carry out with Miss
with
Devyani.
yeah, but because and then if this candidate comes in the middle of the turn, then we cannot give her the turn. It will be in the last because others will be coming at
First, what we can do, you can exchange with Devyani. You can start with Devyani and then let her give the second chance, otherwise at last.
Okay, Miss Devyani, are you here?
Yes, ma'am.
Okay. Okay, so we will start with Ms. Devyani Rajput, and Ms. Devyani Rajput is a research scholar at Department of Pharmaceutical Sciences, Dr. Hari Singh Kaur, Central University, Sagar, Madhya Pradesh, and she will be presenting on the topic pharmacognostic characterization and development of standardization parameters for the quality control of Nictenthus arbotratis linear seeds and leaves. So, Ms. Devyani Rajput, you can please proceed for your presentation.
Okay, ma'am. Please allow me to present my screen.
Now you are co-host.
Okay, ma'am, should I start?
Yes, but if possible, could you please change it to the slideshow?
Yes, ma'am.
It's not in the presentation mode. We can see the slide, but it's not in the presentation mode.
Ma'am, now it is.
Okay, proceed.
Oh, ma'am. Good afternoon, ma'am. This is Devyani Rajput, research scholar from Department of Pharmaceutical Sciences in Dr. Hari Singh Kaur Vishwavidyalaya, Sagar, MP. I am doing my research work under the supervision of Professor Umesh K. Patil sir, Dr. Sunil K. Kasho sir, and Dr. Dharmen Jain sir. Today I am going to present my topic on presentation on pharmacognostic characterization and development of standardization parameters for the quality control of Nictenthus arbotratis lane, seeds and leaf. The objective of my study is to do microscopy of both seed and leaf, microscopy of both physiochemical parameters, phytochemical screening, fluorescence analysis, microchemical test, and elemental analysis. Since we know Ayurveda is the oldest and most widely practiced system of medicine, which incorporates entire plants or their parts, the therapeutic efficacy of drug used in various systems greatly depends on the use of proper and genuine raw materials. Because of these, assurance of safety, quality, and efficacy of medicinal plants and their parts are now very important. Pharmacognostic studies plays a vital role in ensuring the quality, purity, and identification of a plant part or a whole herb plant. The misuse of herbal medicine or natural products
start with wrong identification, which can be solved by pharmacognostic studies of medicinal plants. According to the WHO, the pharmacognostic evaluation of a medicinal plant is the first step to be carried out to establish its identity and purity. Hence, my present study deals with pharmacognostic, physicochemical, and phytochemical studies of seeds and leaves of Nyctanthus arborescens. Here's the plant profile. The plant belongs to kingdom Plantae, clade Trichophytes, order Lamniaceae, family Oleaceae, and genus Nyctanthus arborescens with species arborescens. The bionomial name of plant is Nyctanthus arborescens.
Divyani, your slides are not changing.
So, but in my laptop it is changing.
No, so others are able to see the change in slides?
No, we can't see.
No, you are still on the first slide.
Yes, sir.
Sir, what should I do for that?
Is Miss Devyani, probably it's a fault with your Google Chrome is not updated, but you can't do it now, but you just come out of the meeting and then you enter. Probably it will solve the problem. And if it's not in the presentation mode, in the now it changed. Yeah, but
okay.
Oh, okay.
Should I continue, ma'am?
Yes, we can see now. Okay.
Okay, ma'am, I am on the fourth slide.
Yes, we can see plant profile.
Okay, ma'am. Okay. Now, the common name by which this plant is known is Harshingar, coral jasmine, tree of sorrow, queen of the night, Shefali, Shioli, and a Parijat. This is the morphology of a plant. Nyctanthus arborescens is a large shrub which grows up to 10 meters tall with a quadra-regular branches and flaky grey color bark. The leaves of the plant is 6 to 12 cm long, 2 to 6.5 cm broad. The flowers are arranged at the tip of branches, terminally or in the axials of leaf, and are often seen in clusters of 2 to 7 together. 2 stamens are inserted near the top of the corolla tube and stigma is obscurely bifid. The fruit of the plant are flat, compressed, brown, heart-shaped to round capsules with 2 sections, each containing a single seed. Then the seeds are, the testa of the seeds are thick. Outer layer of large transparent cells is heavily vascularized. Cotyledones are flat and radicle is inferior. The chemical constituents present in leaves of the plant are D-mannitol, beta-citosterol, flavonol glycoside, astragalin, nicotiflorin, olenolic acid, nyctanthic acid, tannic acid, ascorbic acid, and etc. Then flowers are mainly consist of essential oil, nyctanthin, D-mannitol, tannin, and glucose carotenoids. Then seeds are basically consist of arbusticide A and B, glycerides of linoleic, oleic, lignoric, stearic, palmitic, and myristic acids. Then bark is rich in glycosides and alkaloids. Then the plant has various medicinal uses, out of which leaves are used for its use in sciatica, arthritis, fevers, rheumatism, and various painful conditions. Then flower is known for their use in dysphagia, graying of hairs, and baldness, astringent, stomach, and carminatic in nature. Then seeds are known for its use in piles, baldness, scurvy, and hair tonics. Then stem for relieving headaches, and bark for relieving swelling of lungs, and oil of plant and flower are used for its relief to pain of eyes. Oil produced from fragment flowers is used as perfume. Now these are my pharmacognostic characterization of leaves. This is the dorsal view of the leaf and the ventral view of the leaf. I found that the taste and odor of the leaf was characteristic. The upper surface of the leaf was deep green and the lower surface was light green. Texture of the powder was coarse, type was discretely opposite, and the shape of the leaf was oval, margin entire, Apex was found to be acuminate, base was rounded, venation was reticulate, and the length and breadth was 6 to 12 centimeter and 2 to 6.5 centimeter long. The surface was hairy of the leaf. Then I have performed some physiochemical parameters on the leaf, in which I found that the water extractive value was found to be higher than the alcoholic and the ether extractive value. Then the water soluble ash value of the leaf powder was higher than other ash values. Then moisture content was 4%, and the soil index was found to be 0. The forming index was 250, and pH of 1% and 10% solution was found to be 6.25, 5.55. The crude fiber content of the leaf powder was 33%, and the fat content was found to be 6.5%. Then I have performed some fluorescence analysis on the leaf powder, where I have treated my powder with various reagents, in which I found that powder was dark green in daylight. Short UV light shows the dark green color, and a long UV 365 nanometer found to be gray. Then aqueous extract, ether extract, methanol extract was also seen in different wavelengths, in which I found that aqueous extract was brown in color in daylight, and greenish in short UV, and brown in long UV. Protein, mucilages, alluron grains in the leaf powder. Alkaloid to present both ethanol and water, but carbohydrates in leaf surface constant are found that stomata number at upper surface was 0, so that the stomata index was also found to be 0 at upper surface, while in the lower surface it was 337.22, and the index was found to be 14.78%. Then bean islet number and bean termination number was found to be 2.16, 21.9, and the palisade ratio was found to be 7.02.
Miss Medha, we can't see your slides.
Hello?
We can't able to see your slides.
Ma'am, abhi dikh rahi hai?
No, Divyani, it is not appear.
Ma'am, it is showing that host disabled participant screen sharing.
But
Share
how
now.
it, yeah. Yes, it's
now it is
visible.
visible.
Yeah.
Okay, ma'am, should I continue from the current slide or?
No, from the same slide you can continue.
Okay. So these are the leaf surface constant, where I found that the stomach surface.
I think the problem is from the presenter side.
Yes, because we are not able to hear her voice also.
Yes, madam.
I think she has connection problems.
She has a problem and her allotted time also is out, I think.
It's 4:08. I think 4-5 minutes remaining maximum, not more than that. Miss Devyani Rajput. I think she is not here because she's disconnected, I think.
We can ask the first candidate if she's ready.
Yes, that's better. Yes, sir.
Okay. Okay, let me ask because I already she already had problem, but let me ask her. Miss Medha.
Hello.
Yes.
Hello.
Yes. Yes.
Ma'am, can you hear me, ma'am?
Yes, yes, we can hear you now.
Okay, okay, ma'am. Can I go ahead, ma'am?
Yes, yes, you sir will make you co-host, so you will be able to share your slides. She is here now. This is the problem. That's why. Okay.
Okay, ma'am. Let me wait for a while, ma'am.
Okay. Miss Devyani, your connection is very poor, I think. It's like we were not able to hear you also, and it was disconnected in the middle of the presentation. Do you have better connection now?
मैंने अपने phone से connect कर लिया.
Okay. Please continue.
Okay, ma'am. Okay,
you have few minutes only left, 3 to 4 minutes. Please try to complete in 3 to 4 minutes. Okay.
Okay. Okay, ma'am. Okay. Bean Islet number was found to be 2.6, and bean termination was 21.9, and Palisade ratio was 7.02. Then these are some pictures which I have clicked during my leaf surface constant study. Where you can see in first picture stomata and subsidiary cells. Then in second picture palisade cells, and in the third picture bean termination and bean islets. Then this is the microscopy of leaf lamina, where upper epidermis has shown only the straight wall epidermal cells, and the second picture is of lower epidermis, where we can see the stomata cells, which is of anomocytic stomata type. Then this is anomocytic stomata, where we can see a guard stomata pore, subsidiary cells surrounding the stomata, and the guard cells. Then the trichome found in this plant was erect trichome with pointed apex. This is the powder microscopy of leaf, where we can see the flowing basils, part of lamina and section view trichomes, then lower epidermis in different forms. Then palisade cells, epidermal cells, and calcium oxalate. Then this is the TS of leaf section, where we can see the epidermis, parenchyma, scalenchyma cells flowing, and xylem. Then at the lower side we can see the ground tissue and trichomes. In this section we can see the lower and upper epidermis, cuticle, spongy cells, palisade cells, and the glandular trichomes. These are the SAM images of my leaf powder, where we can see non-glandular trichomes, Anemocytic stomata, lower epidermis. This is the elemental composition of my leaf powder, where carbon was found to be in higher compound in a higher amount than silicon, oxygen, and at a very low amount we can see the chloride. Then the pharmacognostic characterization of seed. Here we can see the mesocarp in the seed, epicarp, apical notch, rise like midrib, and the seed of the plant. Then microscopy of seed, the taste and odor was characteristic. Upper color of the seed was reddish brown and the lower color was grayish brown. The texture of the seed was sandy, shape was spheroid, 6 to 12 centimeter, width is 2 to 6.5 centimeter, and surface was rough. Then these are my physicochemical parameters of the seeds, where the water solubility was found to be high and the total H value was found to be high. Then solid index was 2, forming index was 3. Then the crude fiber content was 66.47% and the fat content was 17.55%. This is the fluorescence analysis of the seed. This is microchemical test of the seed.
Veda Paul, can you mute it?
ஏய் வாங்க வாங்க என்னோட பிபிடி யார் வந்து ரெடி பண்ணா
This is microchemical test in which we can see the presence of fixed oil, calcium oxalate, and grains. Then in phytochemical analysis, we can see that alkaloid and flavonoids are present in both ethanol and ethyl acetate extract. Carbohydrates, proteins, and glycosides are only present in ethanol extract. Then terpenoids are only present in the ether extract. This is the powder microscopy of seed, where we can see etesta, cell of mesotesta, oil granules with starch grains, fiber in different stains. Then in microscopy TS of seed, we can see the columnar scleroid of the epidermal cell, cuticle of the seeds, middle part of the seed coat, epidermis, palisade cells in the seed, and parenchyma cell. In SAM images, we have found that exine sculpturing was seen in first picture. Then in second picture, we can see the epidermal cells. In third picture, we can see the tester surface. Then in elemental compositions of seed powder, we have seen that carbon was found to be in higher amount than in oxygen. Then in very least amount, we have seen calcium. Then in conclusion, the standardization of integral parts is correct. Vascular and cup-shaped dialogue leaves of fiber and non-pigment containing cells are the characteristics of the plant. Columnar scleroid was seen in the epidermal cell of the CTS. In the present investigation, consider amount of total ash was noted in leaf and seed, finding then 4 types of ash were determined of the leaf and seed of Nycthanthus rostricus lane, and their percentage value were recorded. Sulfated ash was higher than the total ash, acid insoluble ash, and water soluble ash in the seed and leaf. Extractive values are useful in the determination of exhaustic and adulterant drugs. Extractive values are also useful to evaluate the chemical constituents present in the crude drug and also help in estimation of specific constituents soluble in particular solvents. Elements like calcium, silicon, oxygen, magnesium, aluminum, chloride were found in leaf and seed respectively at concentration in decreasing order as mentioned here.
Okay, Miss Devyani, thank you.
Thank you, ma'am.
Okay, I think although I think all of us has problem in catching your presentation, but somehow I was able to hear better in the last slides of your conclusions and some earlier slides also. but the initial part was not very clear. That is the objective. So can you please correlate your objective with the conclusions?
Okay, ma'am. In objective, I have shown that I have to study my microscopic and different studies of pharmacognostic studies in my research work. So in conclusion, I have found that various different parts in microscopic studies which can be helpful for me to identify my plant, that it is of good quality or of standard quality or of purity.
Okay, okay. And you mentioned in the conclusion about the some ash values, like 4 types of ashes. So you mentioned the sulfated ash is higher than other types. Is there any particular reason that you found out why the sulfated ash was higher?
Yes, ma'am.
And in general, what is the significance of this ash values in the pharmacognostic studies in general?
Ma'am, actually, ash value we used to see ash values because of the extraneous materials which are found in a plant or inorganic type of components of a plant powder. So by performing the ash values, we can see which type of inorganic compound is present in a plant, which can hinder our other further studies like extraction, isolation.
Okay, okay. Okay, now I turn to other chairpersons. Please, any chairpersons for your questions.
So,
Please.
hello Divyani.
Hello,
I have one query. So this is your master's work or research work, PhD research work?
Actually, ma'am, this is preliminary study for my research work.
So what is your future, what is the future study that you have planned? After this standardization, what? Yes, please continue.
Ma'am, standardization of my plant, I am going to do extraction of my plant parts and then the isolation for some single isolates. Then I am going to formulate some formulation for skin cancer disease.
Okay, so you have selected which skin cancer model you will have to use for your future study?
Yes, ma'am, albino mice.
Albino mice.
Yes, ma'am. So it's albino
how will
mice.
you induce the cancer? Skin cancer might be, I don't know, you have know in this time. Okay, that is the most recent part, so let it go. Okay, this is okay from my side. Thank you.
Devyani, I have one question only. Since you know that you are doing this research for your future research.
Yes.
I can see on the same plant, there are various parameters. Everything is done previously by the various researchers. So what is the need of doing by yourself for all the things? You can actually cite their research work if you wish to know about the standardization of the same plant. Because I can see this, the various research on the standardization is going on various part of this plant. And you can go with the skin cancer things that you want, and then you can quote their research work as well. Or do you need to do your own research for that? So I am a little bit confused. Why you need to conduct this study, and then you are saying that this is your standardization, but actually it is done before.
So I have gone through various papers for pharmacognostic studies, but I have found that they have done in 19s. मतलब सर उनके जो पेपर्स थे वो बहुत ही पुराने हैं 1980 और 2000 इतने पुराने पेपर्स हैं and then I have found that they haven't done any SAM imaging नहीं की किसी ने then second thing किसी ने elemental analysis भी नहीं किया हुआ था उसमें.
Okay. Thank you.
Miss Devyani, one more question from my side.
Yes,
which part of the plant you will use for your future study?
So, ma'am, I am going to use seeds and leaves of the plant. for my
Why?
patient birth.
Why?
Ma'am, I have found that ma'am leaf already use हो चुकी हैं, तो मतलब उनका एक्सट्रैक्ट एज अ एंटी कैंसर यूज हो रहा है. तो मैम मेरा बस ये टारगेट है कि ऐसा क्या है उसमें कि मतलब ऐसा कौन सा आइसोलेट है जो कि उसमें इफेक्टिव है एज अ एंटी कैंसर.
But you have already mentioned that the chemical constituent which are present in different parts of the plant, like in seed, flower.
Yes,
You
ma'am.
already mentioned.
Yes, ma'am. पर किसी ने किसी एक पर्टिकुलर आइसोलेट को लेके काम नहीं किया इस कैंसर में.
Okay, so you think that leaf and seeds are more suitable for the anti-cancer activity?
Yes, ma'am.
On the basis of chemical constituent present.
Yes, ma'am.
Flavonoids are there in leaf and seeds?
Yes, ma'am. Flavonoids are present, but my target was for glycosides.
For glycosides. For skin cancer?
Yes, ma'am.
Okay. What is the mechanism of this glycoside for anti-cancer activity?
Ma'am, they are highly antioxidant, anti-inflammatory in nature, which can result in my anti-cancer activity.
Okay, thank you.
Thank you, ma'am.
Okay, any other question?
Nothing, ma'am. We will proceed for the next.
Okay. Thank you, Ms. Devyani again, and we wish a bright future for your further research.
Thank you, ma'am.
Okay, now we move to another participant, that is Ms. Veda Paul, Germany. Ms. Veda, are you here? Ms. Veda Paul, are you here in the meeting?
Hello?
Yes.
Ma'am, can I present my screen?
Wait for a while. Okay, Ms. Veda Paul is a research scholar at Wells Institute of Science, Technology and Advanced Studies, deemed to be University, Chennai, Tamil Nadu, and she will be presenting on the topic clinico-epidemiological investigation among COVID-19 patients admitted at a tertiary care teaching hospital in Chennai. So, Ms. Veda Pal, you can please proceed and please take care of time, because sometimes we have technical difficulties and it may take more time. So please pay attention more toward this request is for all participants. Please pay more attention toward the methods and results so that we can complete within 15 minutes. Okay, proceed.
Good afternoon, everybody. I'm going to present a topic on clinico-epidemiological investigation among COVID-19 patients admitted at a tertiary care teaching hospital in Chennai. Abstract. The background is coronavirus was a disease found in 2019. It is an expanding.
Slide is not moving. Yeah. We can't see your slide is moving. We can see only the front slide only.
1 second, sir.
Okay. Now, please go for the presentation mode first.
Is it visible, sir?
It is visible, but we can see all your slides. Can you go for presentation mode?
Oh, presentation. Sir,
Can you
sir,
share?
am I audible?
You are audible. Can you
Sir,
share the
there is
solution?
a connective issue. 1 second, sir.
Okay. Yeah, now it's okay. We can see that.
Okay. I'll start from the beginning, sir.
Okay.
Good afternoon, everyone. I'm here to present a clinico-epidemiological investigation among COVID-19 patients admitted at a tertiary care teaching hospital in Chennai. Abstract. The background is coronavirus disease. It is 2019. It is expanding worldwide. The characteristics of COVID-19 infections in patients varies from country to country. To move forward, clinical data on infected patients were needed. Methods. This was a retrospective single-center study with 50 patients of data regarding epidemiological, demographic, clinical, and laboratory parameters, management and outcome of COVID-19 patients admitted in the tertiary care teaching hospital in South India. The results, data of 15 patients who were confirmed with RT-PCR for COVID-19 were included in the study. Patients of 60 to 69 age group was affected more due to their immunity status and comorbid conditions. Diabetes and hypertension were considered as major comorbidities. Asthma and CVS were found in severe infected patients who were shifted to ICU after admission. 8% of patients had kidney issues and 5% have only COVID-19 infections. So antibiotics, steroids, insulin, multivitamins, oxygen support, and respiratory physiotherapy were given to patients with respiratory symptoms with or without fever were prescribed with azithromycin. Then the interpretation and conclusion is the characteristic study findings were male gender, older age, and comorbidities, and long time to RT-PCR negativity. The keywords here is clinico-epidemiological study, COVID-19, South India, pandemic situation, and patient care. Introduction. The COVID-19 novel coronavirus disease began to spread in November 2019 in Wuhan, China, starting initially as small clusters of transmission that combined into larger clusters in many countries. Without delay, social isolation, institutional and home quarantine, social distancing, and community containment measures were applied. This study was designed to analyze the psychosocial and socioeconomic and possible environmental crisis based on public perception in India due to outbreak. Shortening the time from symptom onset to isolation is vital and is likely to show the epidemic. So, to reduce the household transmission, strategies are required, which are also useful for supporting home treatment and diagnosis. Peak demand for health services could still be high, and the extent and duration of pre-symptomatic or asymptomatic transmission, if this turns out to be feature of a COVID-19 infection, will determine the success of this strategy. The key activity is to mitigate this epidemic by social distancing interventions, indicating what level of transmission reduction is required. Ongoing data collection and epidemiological analysis are useful, along with essential parts of assessing the impacts of mitigation strategies alongside clinical research. Materials and methods. COVID-19 patients were admitted to the INDO Mission Hospital, Chennai, India from 30 Jan 2021 to 1st March 2021. A focused history including contact history, travel and exposure history, and comorbidities were documented. 50 patients with dyspnea, respiratory rate less than 20 per minute, greater than 20 per minute, or oxygen saturation SpO2 is less than 94% on room had clinical diagnosis of pneumonia, and those deemed to be at risk for severe disease were subjected to chest radiography. Baseline hemogram and liver and kidney function tests were done for all symptomatic patients and those at risk of severe disease. Patients with age greater than 60 years and those with cardiovascular risk factors diabetes mellitus, immunocompromised state, chronic respiratory diseases, and liver or kidney diseases were considered at high risk of progression to serious disease. The patient admitted with COVID-19 were categorized accordingly as per WHO COVID-19 treatment guidelines, depending on the severity of the infection, which is illustrated in the table 1, which is presented in the next slide. All patients were treated symptomatically and were continued on treatment for pre-existing diseases. Patients with respiratory symptoms with or without fever were prescribed with azithromycin. Baseline echocardiogram ECG was performed in all patients as indicated. Oxygen supplementation was given to patients with dyspnea, and patients were discharged after negative RT-PCR test report, along with normal chest examination findings or improvement in chest radiography. Death, discharge, or still admitted were recorded as outcomes. The statistical analysis is, if continuous data were normal, data were presented as mean, standard deviation, and if data were not normal, as median, interquartile range, or frequency and the percentages of category variables. And chi-square test, students t-test, or Mann-Whitney test were used to analyze comparability of groups as appropriate. IBM SPSS statistics version 2.26 were used for statistical analysis. This is the table 1 from WHO classification of COVID-19 infected patients depending upon the severity of the condition. So can we move next slide, sir?
Please go ahead.
The results are a total of 50 patients were considered, of whom 39 were males and 11 were females. Mean age was found to be 61.4 years, and patients of 60 to 69 age group was affected more due to their immunity status and comorbid conditions. The most common exposure characteristics include recent travel history of for past 14 days inside and outside the country and contact with COVID-19 patients. Diabetes mellitus was the most common comorbidity observed, of which 38 patients were hypertensive. And there are no significant difference between a severe disease with respect to disease, gender, smoking status, and alcoholic status. The survival rate of 48 patients who were discharged with improvement was found to be 98%, whereas 2 patients were dead due to treatment failure, was found to have a survival rate of 2% with interquartile range between 36.5 to 13.5. And it is described in table 2. The baseline characteristics of admitted patient with COVID-19. The interpretation and conclusion. The characteristic study findings were male gender, older age, comorbidities, and long time to RT-PCR negativity. Thank you.
Thank you, Ms. Veda. I have few
Thank you,
queries.
sir. Please proceed. Yes.
Yeah. We have few queries regarding your presentation. As you said, it's a retrospective study. So you have a few people in the study. All are male or female also there?
Retrospective, sir.
Yeah, it's a retrospective. I'm asking, all patients are male patients or female patients also there?
Yes, sir.
Epidemiology, is there all male patients you have taken or there are female patients as well? Am I audible to you?
Both patients were admitted. Yes, sir. Yes, sir.
Yeah. Is there any correlation between the sex? Male have the more prevalence or the female have the more prevalence or anything like that? Or any age with severity? We know that there is a comorbid conditions because it's always causing a severity. So,
And the older
have you found anything?
Yes, the older age people among 60 to 70 people were highly affected, sir. And regarding the gender, male was highly affected.
Okay. But in your conclusion, you said something related to the male only. What was it? in the last slide.
Sir, can't audible.
Hello. Am I audible to you?
Hello.
Hello.
Yes, sir. Yes, sir.
Yeah.
Hello. Am I audible, sir?
Yes, you are audible. Very much audible. Can you tell me in the
Okay, sir.
last slide you are telling about only the males? That what I'm confused.
Yes, sir.
Yeah.
Yes, because it may be due to occupation. Male are at high risk, sir.
Okay. Thank you.
Yes, sir.
Please proceed. Sunita ma'am, the other judges may ask the questions.
Yeah, other judges, please, if you have any questions.
Ma'am, as we are running short of time, we can proceed next, ma'am.
Okay. Okay, Ms. Mehta, thank you very much for your participation.
Thank you, ma'am.
Miss Megha Joshi is here. Miss Megha Joshi.
Yes, ma'am.
Okay. Miss Megha Joshi is a research scholar at Department of Pharmaceutical Sciences and Drug Research, Punjab University, Patiala, Punjab. And she will be presenting on the topic solidified self-nano-emulsifying system of itraconazole, formulation, optimization, and characterization. Ms. Megha Joshi, you can please proceed for your presentation.
Okay. Okay, my screen is visible.
Yes, yes.
Okay, good afternoon to one and all. My name is Megha and my topic for oral presentation is solidified self-nano-emulsifying system of itraconazole, formulation, optimization, and characterization. Okay, so firstly, what is fungal infection? Okay, fungal infections remains a significant cause of morbidity that since 1980, only 2 of the 3 major classes of antifungal drugs, azoles and polyenes, are in current use, and the third one is styronocandins, has been recently discovered. Spectrum antifungal agents with an acceptable safety profile and with both intravenous and oral formulations are available at this time. Okay, so this is candida infection. Then my formulation is SNADS. What are SNADS? SNADS is a mixture of oils, surfactants and co-surfactants which are emulsified in aqueous media under conditions of gentle agitation of digestive motility that would be encountered in the gastrointestinal tract. Okay, so the aim and objective of my study is the present study was aimed at developing solid self nano emulsifying drug delivery system of itraconazole to an invasive fungal activity.
Ms. Megha,
Yes, sir.
your voice is not coming in between. Is it with me only, or all have the problem?
No, it's breaking. Yeah.
My voice is not, maybe due to network issue. Now it is audible.
Yes, now it is audible. Once again, no audio.
Megha, we cannot hear you.
Okay, ma'am, now my voice is audible.
Yes, but yes, please continue.
Ma'am, my voice is audible.
Yes, yes, please start.
Okay, ma'am. Ma'am, so why shall I start from here only?
Yes, you can start from there only.
Okay. So next is a method, and it was found to be 167 degrees Celsius, which is in accordance with the entropy was done, and the peak observed was similar to the peaks that are reported in the literature. So then the method validation was done. Firstly, the standard plot on UV spectrophoto on of different method development, the linearity range, the intercept, the LOD, LOQ. Then after that, there is a formulation developed. Hello. कर रही हूँ मैम मैं. Okay, okay. Expectants. Selected for further study. Then there is a solubility of itraconazole in oil and S-mix mixture. Containing the formulation 100 mg. Of pseudo-ternary phase diagram. formulations and 2:1 ratio was selected as a combination of surfactant and co-surfactant because it shows the maximum microemulsion area. And this is the pseudo-ternary phase diagram of that. Then after that, self-emulsification and dispersibility studies were performed. And this is the formulation that shows the clarity and has a robustness of dilution that will be selected for the further optimized formulation, that is 45.28 nanometer, and PDI was 0.436. And this is the zeta potential of the optimized formulation, that is minus 25.2 millivolt. Then characterization and evaluation of solid SNEDs. Then for the maximum oil adsorbing and desorbing capacity. Different that is pure drug, the adsorbent, the physical mixture of drug and adsorbent, and solid SNEDs. And as we can see in the graph, the solid SNEDs, there is a disappearance of the peaks in the solid SNEDs that revealed that expression pattern of solid SNEDs. There is a pores in case of solid neosilin US2. So solid SNED shows the drug release of more than 95% in 15 minutes. Then stability studies. To conduct the stability studies, solid SNEDs were incorporated into hard gelatin capsules. Further, the capsules were stored in HDPE bottles and kept in stability chamber for different time intervals. Thereafter, drug content and drug release of the capsules were assessed out. So after the different time interval, there is no major difference in the in vitro of
Excuse me, Megha. Megha, your voice is so much breaking. I can't hear you.
Sunita ma'am.
Yes, it's all almost anything we were not able to hear. Yes, I was just saying her slide was moving, that's all. Megha? I think each presenter has problem this time. And now she is not able to even move the slides. Megha, we cannot hear you.
You might have the issue with the mic. I think so.
She is presenting. We can see her, but
Anyhow,
there is a
complete
lot.
that because she can't rectify this. It seems to be a permanent
But
issue.
she is not able to move the slides and probably can't hear us. I don't know. She is not hearing to us also.
Anyhow, evaluate accordingly because we are tight of time.
Time is also over.
How many minutes are remaining?
Take 1 minute only, 2:21 it should be completed. It's now 2:21.
Her slides is also in something different mode. See, it's like in edit mode.
Yeah, presentation.
Megha, can you hear us?
Yes, ma'am.
But we had a lot of problem and we cannot hear like almost anything what you were presenting, and now you are stuck on this slide since 5 minutes in vivo standard plot.
Yes, ma'am.
Are
Now
you
my
moving
slide
the
is
slides?
moving. Now my slide is moving.
Now
Yeah.
it is moving, but last 5 to 7 minutes we didn't hear you anything. Okay, you can just continue and complete. Okay.
You just conclude this now. You just come on the conclusion slide.
No, let her allow to discuss the pharmacokinetic. Just be fast, 2 to 3 minutes. Okay.
Okay, ma'am. Okay, so I have made the formulation that is solid SNADs, and this is the pharmacokinetic studies.
She has the mic issue.
Yes, again.
And then solid SNADs, that is it shows the Tmax of 2 hours, and marketed formulation that shows 4 hours. And this is the Cmax that is also more in case of solid SNADs, that is 203. Shows that the solubility and oral bioavailability of it.
Megha, am I audible to you? Megha, we are not able to hear you. You are not audible.
If time is complete, then we can move next, I think. So there is no use if she is unable to speak.
Yes,
Sir, what's
her
about
time is
question answer
complete.
session?
But the problem is she will not hear us.
Then what to do about?
She may be hearing us, but she is not able to speak.
Then she can answer in chat box.
Yes, she can use it, but
I think we can ask 1-2 question and then she can explain in chat box so that we can give some numbers to
okay. Now I think she wrote in the
Yes.
chat box. So please
Yes.
proceed, Chairperson. If you have any question, please proceed.
Megha, you come on the standard plot, the slide of standard plot, HPLC.
Yes.
Yes. So this is the plot between area and concentration in nanogram per ml. Why did you not plot this curve from 0, 0? It is not started with 0, 0. No, it's not because of linearity. You are making a line equation by plotting this graph, but it should be started with 0, 0. Okay, from my side, it's done.
Okay, other chairpersons?
It's okay from our side, ma'am.
Okay, Megha, have you solidified the systems after preparation? You mentioned that it is solidified system. What was the method used for solidification? Okay, which method? Adsorption technique. Okay. Okay, then. If no other question, then we can move. Thank you very much, Megha. Although we have problem, but it was a nice work. Okay, thank you for your participation. Okay.
Ma'am, continue with the next one.
Oh, sorry. I already started. Miss Ginny Christian.
Yes, ma'am.
Okay. Miss Jenny Christian is Assistant Professor, Department of Quality Assurance, Faculty of Pharmacy, Dharam Singh Desai University, Nadiad, Gujarat, and she will be presenting on the topic stability indicating HPLC method development for simultaneous estimation of glycopyrrolate and formoterol fumarate. Miss Jenny Christian, you can please present now.
Yes, ma'am. Is my screen visible, ma'am?
Yes, but not in the presentation mode.
Yes, ma'am. Yes, ma'am. Is it okay now?
Yes, it is good now. Please proceed.
Okay. Good afternoon.
Today, I'm going to discuss on stability indicating HPLC method, development and validation for simultaneous estimation of glycopyrrolate and formoterol fumarate. This is going to be the roadmap of my presentation today. As we know that COPD, that is chronic obstructive pulmonary diseases, it is the third leading cause of death by 2030, will be the third leading cause of death by 2030 as suggested by WHO. And if a person has COPD, then it becomes, for a person, he becomes more prone towards developing an active TB. And so many drugs are approved for the treatment of COPD, and one such FDA approved combination is glycopyrrolate and formoterol fumarate, which is indicated for the long-term maintenance and treatment of COPD, including chronic bronchitis or emphysema. Looking towards the drug profile, formoterol fumarate, it is beta-2 adenoceptor agonist, and glycopyrrolate, it binds competitively to the muscarinic acetylcholine receptor, and which inhibits the action of acetylcholine. So on reviewing the literature, it was noticed that individually as well as in combination of these drugs with other drugs, so many methods like different chromatographic methods and UV spectrophotometric methods have been reported. If I can share my screen and show you the literature review for literature review, just give me a minute, I'll be sharing. Okay. Can you see my screen? Presentation mode?
Yes.
Yes. Okay, somehow I'm not able to open that PDF in which I have summarized all different chromatographic and spectroscopic different analytical methods which are individually done for formoterol fumarate and individually done for glycopyrrolate, as well as combination of these drugs with other drugs. If time permits at the last, I'll be showing you that PDF. And talking about the literature review for both of the drugs regarding their analytical method development for glycopyrrolate and formoterol fumarate. So we have developed literature review reveals that there is a method which is reported on UV spectrophotometric. We have HPLC method, but you can see the retention time, they are very nearer, which is not suitable to study the stability study of drugs. Similarly, there is one article where they have done different methods. So HPLC method using ion pair chromatographic methods. So on literature review, I concluded that there is a need to develop a method which can be applied to study the stability studies of the drug as well as to study the marketed formulation. So this is my work done starting with the identification studies. So formoterol fumarate and glycopyrrolate were procured from Zydus and Intas respectively. All the chemicals and reagents used were of HPLC grade. For identification study, melting point study was done, UV absorption study was done, IR was done. In UV absorption study, as you can see in the graph, that the lambda max of formoterol fumarate was observed to be at 212, while for glycopyrrolate, no specific peak or a curve was obtained, but it was showing an absorption from 236 to 200 nanometer. And this zero order spectra were similar to the reported spectra in various research articles, confirming the identity of formoterol fumarate and glycopyrrolate, while melting point studies also confirmed their identity. Talking about the solubility, formoterol fumarate, it is having very slight solubility in the water, and glycopyrrolate is freely soluble in water. IR spectroscopy was done where the sample and the reference spectra were compared, which were overlapping, confirming the identity of the drugs. This is the sample spectra of formoterol fumarate, and this is the standard spectra taken from in pharmacopoeia of formoterol fumarate. This is the sample spectra of glycopyrrolate, which exactly matches the standard spectra of glycopyrrolate. Mass spectroscopy was done. This is the mass spectra for formoterol fumarate. As we can see that the parent peak was observed at the M/Z ratio of 345, and the peak which is seen as which is seen at 149 M/Z ratio was because of the nitrogen protonation of the ethanolamine side chain, and the M/Z ratio 121. It is because of that 4-methoxytoluene lacking one hydrogen atom, and the peak at 327 was because of the water molecule release, and from that from that 345 M/Z ratio, and 179 is because of the removal of water molecule, and 4-methoxyphenyl propene removal from the precursor ion. So this also confirmed the identity of the received drug, and this is the mass spectra for glycopyrrolate. As you can see that the parent peak was observed at 318.4, which is the molecular weight of glycopyrrolate, and 116 peak. As you can see, this fragment was getting fragmented, and we have the peak at 116 M/Z ratio. So this was the identification studies. Moving towards the HPLC method development, where the Shimadzu model was used for the development. Solutions were prepared where phosphate buffer was prepared using potassium dihydrogen orthophosphate, and mobile phase was prepared. Standard stock solutions were prepared in the mobile phase, where the dilution of where the standard stock solutions were of 1000 microgram for formoterol fumarate and 2000 microgram for glycopyrrolate, as you can read in the slides. And then the optimization was done. So first step in optimization was the selection of analytical wavelength. So 230 nanometer was selected for the analysis of both the drugs simultaneously. Optimization started with taking the ratio of water is to methanol 10:9, which is visible that both the peaks, that means both of them were exactly eluting at the same retention time, plus the peak shape were also not good. Again, the ratio was changed, but still the peak shape was not good, plus the resolution was also poor. So now methanol was shifted, was replaced by acetonitride. So here a peak shape of one drug was good, but the another drug was not good. Peak tailing was observed in formoterol fumarate. So now buffer was tried, and at pH 6, the peak shape of glycopyrrolate, it is not good. Plus the resolution was greater than 2, but the peak shape was not good. And hence the pH was tried to reduce. Initially pH 6 was used, and then pH 5. And then pH 4 gave us good result, but the resolution was lesser than 2. And then again, different combinations of methanol and phosphate buffer was tried, and ultimately the final mobile phase was optimized to 0.05 molar phosphate buffer having pH 3.5 and methanol in the ratio of 30:70, where the retention time for formoterol fumarate was 5.33 and for glycopyrrolate it was 3.6 minutes. Which was having the good system suitability parameter as well. So these are the optimized chromatographic conditions having acceptable system suitability parameters. Method was validated in terms of linearity, where linearity was confirmed having the good R-square value nearer to 1. The linearity range for formoterol fumarate was 5 to 15 microgram per ml, and for glycopyrrolate it was 10 to 30 microgram per ml. Specificity was ascertained by extracting the drug from the formulation and having their spectra and and having their chromatograms. And as you can see that no extra peaks were observed at the retention time of that respective drug, confirming that the method was specific enough to quantify the drugs from the formulation. In terms of LOD, the LOD and LOQ of the method was relatively lower, suggesting the sensitivity of the method. The LOD of FF and GP was 0.430 and 0.772 microgram per ml, and for FF and GP, the LOQ was respectively 1. And 2.187 microgram per ml. Inter-day and intra-day precision, both of them were having the RST lesser than 2%, indicating the precision of the method. In accuracy as well, for both of the drug, percentage recovery was within the range, ascertaining the accuracy of the developed method. To study the robustness, fraction factorial design was applied. That is a 4-factor half fractional factorial design was applied, 2:4 minus 1. Where 2 levels were taken, low and high, and 4 factors were there. The 4 factors were wavelength, flow rate, pH, and volume. Factor code is given A, B, C, D, and high level and low level are written here. So this was the design domain which was generated, and these are the responses which were observed on applying the statistics. The model p-value for the retention time of formoterol fumarate and for the retention time of glycopyrrolate, it was found to be significant. That means the method was not robust in terms of retention time of the drugs, while percentage recoveries of both the drugs were not affected, as the model turned out to be not significant. The adequate precision values was also good, that it needs to be greater than 4, which is an indicative of signal to noise ratio. So that was also good. R-square value was also good, but the model came significant for the retention time of the drugs. And this is the Pareto chart, which shows that the factor D, which was methanol percentage, was a very critical factor affecting the retention time of both the drugs. And this is the 3D response surface plot. As you can see that when the methanol concentration is increased from 65 to 75%, there is increase in the value of retention time. And similarly for, this is the graph for formoterol glycopyrrolate, and this is the graph for formoterol. Synthetic mixture analysis was done, and the method gave a good assay result. Yes.
हाँ जी बताइए.
Hello.
नागा जी, हाँ how is
Excuse
student
me,
का है मैंने उसको अभी डिटेल
can you
भेजी
please mute yourself? Excuse me. Excuse me.
Should I continue?
Yes, yes, please continue.
So analysis of synthetic mixture, it was done. In analysis of synthetic mixture, the results obtained were good and RST also was lesser than 2, as you can see the chromatogram, and hence the method can be successfully applied for the quantification of drug in their marketed formulation. Next, the method was applied to study the stability of drugs in various conditions. First, acid degradation, where 0.1 normal HCl was used, and the sample was kept in contact with the acid for 4.5 hours at room temperature. And the degradation observed for glycopyrrolate was 10%, and for formoterol fumarate it was 12. That's approximately 13%. Similarly, for base degradation, 0.1 normal NaOH was kept in contact with the drug for 5 hours at room temperature, and the degradation observed was 16% for glycopyrrolate and 13.4% for formoterol fumarate. For oxidative degradation, also 1 ml of 3% hydrogen peroxide was used, and the sample was kept in contact with the hydrogen peroxide for 4 hours at room temperature, where the degradation observed for glycopyrrolate was 10%, and for formoterol fumarate it was 16%. Thermal degradation also, where the drugs were exposed to a temperature of 105 degrees Celsius for 4 hours in an oven, and then the final dilution was made in the concentration range which falls in the linearity that gave the degradation of glycopyrrolate 12.8 and formoterol fumarate 15.9. For photolytic degradation, the samples were exposed to the sunlight for 3.5 hours, and then the concentrations were diluted to have the range in the linearity range, where the glycopyrrolate shows 7.885% degradation and formoterol showed 11.7% degradation. In summary, glycopyrrolate was showing the highest degradation in the basic medium, and formoterol fumarate was showing the highest degradation in the oxidation medium, while glycopyrrolate was observed to be least degrade in thermal condition, and formoterol fumarate was observed to be least degraded in the thermal conditions as well. So a simple, accurate, sensitive, and a precise HPLC method was developed for the simultaneous estimation of the drugs, that is formoterol fumarate and glycopyrrolate, and it was validated where all the validation parameters were within the range. Plus, robustness study was done where percentage recovery and resolution changes, they were not significantly affected by the parameters which were taken. Only for the retention time, the methanol content was found to be significantly affecting, and therefore, while taking the method into application, a strict control is required on methanol concentration. And the method was successfully applied to study the effect of stress stability conditions where the drug peaks were obtained without any interference of degradant. Hence, the method was developed and validated, and it can be successfully used for the quantification of drug in marketed formulation and to study their stabilities for routine quality control analysis. Thank you. These are the references. I've concluded, madam.
Okay, thank you. Thank you very much. Miss Jenny, is this work published?
No, it has yet to be published. The article is under process.
Okay, okay. Chairpersons, please proceed if you have any questions.
Miss Jenny, you have estimated simultaneously both the drugs.
Yes.
And you have developed a method, and also you have done the stability studies.
Yes.
Okay. But as you have concluded that both the drugs are individually estimated by RPHPLC method, but no one method is available for the simultaneous estimation of both the drugs.
No,
but
yes, ma'am, continue.
yeah. But I can see that there is a method, there are 2-3 methods are available for the estimation, simultaneous estimation of both the drugs.
Yes, ma'am. I have shown that also in my presentation that 3-4 methods are available regarding UV, as well as a method is reported to study the HPLC for the stability of study of the drugs. And I have mentioned that in my slide also that the methods are reported for their simultaneous estimation, plus an article is also reported for their simultaneous estimation by HPLC and they have applied it in stability study. But when I when I tried using that same mobile phase, the results are not reproducible. Also, the retention time which they have mentioned, it is very nearer 2.3 and somewhere, right? So which I found that it is not good to study the stability, because you have so many degradant, and it will not be, that means you need to have a good resolution, plus at least if you can have the pics beyond 3 minutes, it will be good. So that's why I have developed the method.
Okay, okay. What is your mobile phase?
My mobile phase is phosphate buffer, pH 3.5, and methanol in the ratio of 30:70.
Okay, okay. In that paper, they have taken water and acetonitrile in 50-50 ratio.
Yes.
Okay, okay, good.
Okay, thank you.
Any other question? Okay, if no, then we can proceed for the next presentation. And Miss Jenny Christian, thank you very much for your participation and sharing your research with us. Thank you.
Thank you very much, ma'am. Thank you.
Okay, we have next, okay, Miss Uma Rajeshwari is the next presentee, and I think she is here.
Yes, madam.
Okay. Miss Uma Rajeshwari is a senior research fellow at Indian Institute of Chemical Technology, Hyderabad, Telangana, and she will be presenting on the topic exploration of anti-inflammatory potential of microbial derived 1,O-methyl chrysophenol from Amycoleptopsis thermoflava MTCC 25037. Miss Uma Rajeshwari, you can please now proceed for your presentation.
Okay, madam. Is it visible, madam, my screen?
Yes, yes, it's good.
Can I continue?
Yes, yes, please proceed.
A very good afternoon to one and all. This is Uma Rajeshwari. So I'm working as a SRF from ICT. So today I'm here to present my research work, exploration of anti-inflammatory potential of microbial derived 1,O-methyl chrysophenol from Amycoleptopsis thermoflava. So these are the contents of my work. So first, before going to my topic, let me explain a brief introduction about inflammation. So actually, basically, physiologically, inflammation is a protective mechanism which will protect us from infections, tissue damage, and cell death. But it associates with a homeostatic balance between regulatory mechanisms, which can switch from pro-inflammatory mediators to anti-inflammatory mediators to do the tissue repair. But if there is any imbalance between these pro-inflammatory mediators and anti-inflammatory mediators, it will lead to a significant cause of mental and physical health problems. And nowadays, chronic inflammation is the leading cause of world's leading cause of death in so many inflammatory diseases like stroke, ischemic heart disease, cancer, non-alcoholic fatty liver disease, autoimmune and neurodegenerative diseases, including recent pandemic COVID-19 also. And acute inflammation develops gouty arthritis in the joints, which is later progressed as a chronic condition. Because of the association of inflammation with these serious inflammatory diseases, we direct our research one more, our motto is to isolate a novel bioactive compound from the natural source, which will have a very few side effects and potent anti-inflammatory activity. Because present nowadays, the existing currently using non-steroidal anti-inflammatory drugs are having severe side effects like liver damage and some allergic reactions. So because of that, we have focused our research in isolation of anti-inflammatory drugs from natural sources. So as a part of our research work, we have chosen the microbial source for isolation of bioactive compounds. So here, more particularly, actinomycetes we have chosen. Why? Because we all know that. So presently in the market, almost most of the antibiotics we have isolated from the actinomycetes only. And not only antibiotics, so many bioactive compounds have been isolated from the actinomycetes. We can call them as a actinobacteria also. So in our present work, we have isolated hydroxyanthraquinones. So these have been well known for their significant anti-inflammatory potential since long ago. So here we have isolated one of the derivative of chrysophenol, which is already having the anti-inflammatory potential. So these are the aim and objectives of my work. First, we have isolated the 1,4-methyl chrysophenol from Amycolaptosis thermoflava, and after that we have screened that compound in docking in silico analysis, and after that we have done the in vitro studies and in vivo studies, as well as histopathology and immunohistochemistry studies. So this is the schematic representation of production and purification of my compound. I can shortly call as a OMC, 1,4-methyl chrysophenol from Amycolaptosis thermoflava. So actually this is the rare actinobacteria. This has been isolated by our lab group earlier from the soil, which is collected from the Medak district. So this actinobacteria is the gram-positive rod-shaped aerobic spore-forming bacteria. So first we have grown that culture, and the method to isolate this compound was already developed by our lab, our colleague researchers. First we have grown the plate culture of this bacteria, and after that we have inoculated that culture into broth media, production media, and we have done the fermentation process for a period of 7 days. After that, from the supernatant, we have extracted our compound by using resin, because our compound will attach to the resin. After that, we have re-extracted resin binded compound into the methanol, and then we rota evaporated and the crude extract was formed. Then we have done the TLC analysis. So in our TLC analysis, from our crude extract, our compound has been observed as a UV active single spot. And after that, we have purified the crude extract by using column chromatography. So this is the TLC chromatogram of our compound, and we have used a mobile phase composition of methanol and chloroform in both TLC and column chromatography also. These are some of the characterization techniques which I have followed for the characterization of my pure compound. So HRMS spectrum, we have observed the molecular formula, and in FTIR spectrum we have identified the OH groups and carbonyl groups, and in NMR and C13 NMR spectra we have identified the structure of our compound as a 1,4-methyl chrysophenol, and IUPAC name was 1-orthomethoxy, 1-methoxy, 3-methyl, 8-hydroxyanthraquinone. So after characterization of my compound, first we have done the screening studies by using in silico docking analysis. Here, first we have chosen the anti-inflammatory enzyme like COX-2, cyclooxygenase 2, which is the which is majorly responsible for the inflammation process. Here we have used a COX-2, COX-2 protein, and ligand is our OMC. And we have used the Moligro virtual docker. These are the binding affinity studies between our OMC and COX-2. And from this docking analysis, our compound has shown a good binding energy with the COX-2, which has been represented by the Mol-Dock score of -99. The more the negative value, the stronger the binding affinity. As well as that, our compound has shown 2 hydrogen bond interactions with the active site of the COX-2, and the more the hydrogen bonds, more will be the binding affinity. And the serine 530 amino acid which is present in the active site is the important amino acid which is responsible for the anti-inflammatory activity, which is already proven by earlier studies. So here also our compound has have a ligand protein interaction with the serine 530 amino acid in the active site. So after in silico analysis, I have progressed into in vitro anti-inflammatory activity by protein denaturation method. So here, what is the principle of this method is actually during the inflammation process, along with inflammatory mediators, some oxidative stress will also be generated. This oxidative stress will lead to the denaturation of some of the inflammatory proteins, which will do the tissue repair. So, if there is a effect of our compound is there on the inhibition of protein denaturation, we can use it as an anti-inflammatory agent. So here in this method, this is the in vitro method. We have used the bovine serum albumin as a protein, and we have prepared 1% bovine serum albumin with sodium phosphate buffer. pH is 0.4. After that, this protein we have treated with the test sample at different concentrations, and we have used indomethacin as a standard drug. And the reaction mixture was incubated at 37 degrees centigrade for 20 minutes and then heated to 95 degrees centigrade for 20 minutes. After this procedure, the turbidity will be observed. So and it was measured by UV spectrophotometer at 660 nanometer, and the percentage of inhibition was measured by using the following formula. So from these studies, the results concluded that our OMC compound has a significant inhibition of protein denaturation with an IC50 value of 67.37 microgram per ml, which is comparable to indomethacin, which is the standard. Coming to in vivo anti-inflammatory activity. So after the in vitro anti-inflammatory activity, we continued our studies to in vivo studies also. All our experiments were performed according to guidelines issued by Animal Ethical Committee at the ISCT. And we have collected adult male Vistar rats of 160 to 200 grams weight. And the method what we have used is carrageenan-induced rat pie edema model. This is the basic method and simple method. That's why we have chosen this method. So the power volumes before and after treatment were measured by using this digital plethysmometer. Actually, the principle of this method is first we need to induce the edema by using 0.1% carrageenan. Then we need to treat our groups with the drugs. Actually, this is the preventive method. First, before induction of the inflammation only, we should treat our treatment groups with our drug. Then we need to induce the inflammation by using 0.1% carrageenan. So this is the experimental protocol. We have taken 5 groups. Group 1 is vehicle control. Only we have injected with saline. Group 2 is positive control with carrageenan induction. Group 3 is carrageenan plus standard drug indomethacin, 10 mg per kg body weight. Group 4 is carrageenan plus OMC, 100 mg per kg body. And so the results of this carrageenan-induced method has shown that our compound has shown a 40% inhibition in the pie edema volume, which is comparable to indomethacin, which has shown the 52% inhibition in the pie edema value. The results were our based results were presented in the graphs. We have analyzed these values by using statistical analysis using one-way ANOVA by Dunn's multiple comparison test. The values were significant with p less than 0.01. So after the carrageenan-induced model, we have used the same tissue samples for the histopathological studies also. In the histopathological studies, what we have observed is what is the edema level and what is the acute inflammatory lymphocytes level in the tissues. So here, coming to the results, then these are the tissue sections, stained sections by using H&E stain. Here, the first vehicle control, there is no edema and inflammatory lymphocytes. It has shown a clear epidermis, dermis, and subepidermis layers. But in carrageenan-induced model, there is a moderate infiltration of lymphocytes and there is a edema. Inflammation was observed in the tissue samples. And then coming to the indomethacin treated group, the number of lymphocytes level was decreased and inflammatory score was also reduced. And then coming to carrageenan plus OMC group, So here also, compared to indomethacin, the inflammatory lymphocytes level was decreased like indomethacin, but inflammation was not that much reduced like indomethacin. This might be due to actually COX2, our ligand is inhibiting the COX2. So COX2 is the major enzyme which is expressed in the lymphocytes. So maybe because of this inhibition of COX2, it is reducing the inflammatory cells number compared to the inflammation process. So after the histopathological studies, mainly histopathological studies, in histopathological studies, we can observe the pathology and morphology of the tissue. And in immunohistochemistry, what we can observe is this is the diagnostic method which can observe the target molecules levels in the tissues. Here, target molecules means I already told you that inflammation is a homeostatic balance between pro-inflammatory mediators and anti-inflammatory mediators. So during the inflammation process, pro-inflammatory mediators will be released and anti-inflammatory mediators.
What next?
Hello? Hello?
Please, you can proceed.
You can please continue.
So pro-inflammatory mediators and anti-inflammatory mediators both will be released. So in our present study, we selected some pro-inflammatory mediators and anti-inflammatory mediators, and we have checked their levels in the diseased group and treatment group. So actually here, what are the pro-inflammatory mediators we have chosen are CD80, CD31 and CD68. And anti-inflammatory mediator, what we have chosen is arginase. Coming to the role of arginase 1. Arginase 1 is the anti-inflammatory mediator which can produce ornithine during the inflammation process. That ornithine will involve in the tissue repair process. But CD31, CD68 and CD80 are the inflammatory molecules. So CD80 is a pro-stimulatory molecule which is released during the T cell activation and during the inflammation process. CD31 is the endothelial marker which can be overexpressed during the inflammation process. CD68 is the macrophage which is highly upregulated during the inflammation process. So coming to results of our study. Here in our study, we have checked the levels of these molecules in treated groups, both in indomethacin treated and OMC treated, along with our carrageenan treated positive control group. So coming to arginase levels, arginase levels were highly expressed in the OMC and indomethacin. They were very low in the carrageenan treated group. It indicates that our compound has increased the anti-inflammatory markers so during its anti-inflammatory mechanism. But coming to CD80 and CD68 levels, our compound and indomethacin comparably they have reduced the levels of CD80 and CD68 in our treated groups compared to the disease control. Coming to the CD31 also upregulated in the disease process, disease group, and it was downregulated in the indomethacin and OMC group. So these results indicate that our compound is showing significant showing the significant anti-inflammatory activity along with showing its release of more anti-inflammatory mediators during the inflammation process. So we have compared our results by using statistically, we have compared and we have analyzed by using one-way ANOVA. We have compared both inflammatory cell score and edema score in the histopathology studies. And in immunohistochemistry studies also, we have compared the levels of arginase 1 and CD80, CD31 and CD68 by using ANOVA. So conclusion is a derivative. Our compound is a derivative of chrysophenol, which has been isolated from Amycoleptosis thermophilova. It has shown a significant anti-inflammatory activity both in vitro and in vivo models. So in future we are planning to do our study in a specific inflammatory diseases like gouty arthritis. In a cell lines models also, we are planning to do our anti-inflammatory activity by using our isolated compound. Thank you, sir.
Thank you, Ms. Uma Rajeshwari. It's an elaborative presentation, and I welcome any chairperson for any questions.
So, hello, Uma. Am I audible?
Yes, madam.
So you have presented a very good work. It's a very vast and compiled study, and you presented in a very good manner. So I have just one query. You have showed the OMC, that is the extract, it showed the higher therapeutic efficacy, higher anti-inflammatory activity as compared to the standard. So can
no, madam, not more. It is comparable to. It is showing more activity comparable to indomethacin, not more than indomethacin.
Okay, so what is the extent? How much extent it showed the edge compared to the indomethacin?
Actually, 70%, more than 70% activity it has shown comparable to the indomethacin.
Okay. Good.
Thank you, madam.
Any other question?
Nothing, ma'am. We will proceed it.
Okay, thank you.
Thank you, madam. Thank you very much.
We have.
Can I stop my screen sharing?
Yes, you can unpresent. Dr. Jehad Syed, are you here?
Yeah, good afternoon, ma'am.
Okay. Dr. Jehad Syed is research scholar in the Department of Pharmacy Practice, JSS College of Pharmacy, JSS Academy of Higher Education and Research, Mysuru, Karnataka, and she will be presenting on the topic assessment of medication adherence and beliefs about medicines among the elderly. So, Ms. Jehad Syed, you can please proceed now for your presentation.
Okay, ma'am. Thank you. Is my screen visible, ma'am?
Yes, it is.
Okay. Thank you. Good afternoon, everyone. Myself, Dr. Jehad Syed, research scholar, JSS College of Pharmacy, Mysuru. Today, my topic that I'm going to present is assessment of medication adherence and beliefs about medicine amongst the elderly. So to introduce my topic, it is estimated that approximately 10% of the hospitalizations 10% of the hospitalizations are related to medication non-adherence, which contributes to an estimated avoidable healthcare cost of $300 billion USD in the United States alone. With the increase in age, chronic diseases become more prevalent, and increased volume and complexity of medication regimens leads to an increased risk of medication non-adherence. Again, other factors also play a role in increasing the likelihood of medication non-adherence, which include polypharmacy, decreased visibility, motor dexterity, and decreased autonomy. Medication non-adherence may be associated with poor therapeutic outcomes, disease progression, increased healthcare utilization, and an estimated burden of billions per year in avoidable direct health cost. Non-adherence may be attributed to a set of behaviors, including both intentional and unintentional causes. Unintentional and non-adherence refers to a non-deliberative alteration in treatment, which could be due to forgetfulness, poor manual dexterity, losing the medications, and economic reasons. On the other hand, the intentional non-adherence is the deliberate alteration in the treatment, and which is largely associated with lack of patient motivation. The patient's belief about the necessity of taking their medicines and concerns regarding their medicines have shown to predict the patient's adherence to their medicines. A strong belief in the necessity or concern regarding the medicines is associated with a higher adherence rate. Therefore, it is essential to assess the medication belief to understand the adherence behavior among chronic medication users. The objective is to assess the medication adherence and the beliefs about medicines amongst the elderly. Coming to the methods, the study was conducted at the Department of Geriatrics in a tertiary care hospital, and the study design being a prospective cross-sectional study, and the study period was conducted over a period of 1 month. The study participants were all elderly patients who are equal to or more than 60 years of age, admitted to the inpatient wards of the geriatrics department in a tertiary care hospital. The sources of data were, all relevant data was collected from medical records, patient case notes, patient and patient caretaker interview. So the study tools were simplified medication adherence questionnaire, beliefs about medicines questionnaire. The data thus collected were categorically analyzed, and the association between the gender, literacy, the number of comorbidities, the level of adherence was tested using chi-square test, and man-made knee test was used to find the relationship between the adherent and non-adherent groups and their beliefs about medicines. So coming to the results, the admissions in the month of December 2021 were 55, and a total of 47 cases were reviewed by the clinical pharmacist, of which 8 had no comorbidities. So they are not chronic medication users, so they were excluded. Then department transfer, they were transferred to other departments from geriatrics, and they had less than 24 hours admission, and they were 2 were critically ill, so we couldn't question the patient regarding their medication beliefs, and 2 had incomplete details, and 1 death was reported. So totally 30 patients were included into the study. So coming to the social demographic details, a total of 30 patients were interviewed during the December 2021, of which 70% were males, and the average age was found to be 72 years, and the average length of hospital stay was found to be 10 days, and the medication adherence was reported among these study participants was found to be 50%. Majority of the study participants endorsed the belief that their current health depended on their medications, that is 80% of them, and 73% of them believed that their medications kept them from getting worse. Few concerns were also reported, such as 86% of them were worried about the long-term effects of their medications. Also, 23% mentioned that the doctors use too many medicines, and 30% of them also believe that medications are addictive. So these are the responses. With the health, is my health dependent on my medicines? 66% of them agreed, with majority being uncertain, saying that life would be impossible without my medicines. 46% mentioned that without my medicines, I would be very ill. And 50% of them agreed that their health in the future will depend on their medicines. 63% of them agreed that the medicines protected them from becoming worse. Regarding the concerns, 36% of them had agreed and disagreed regarding that taking medicines worries me. 23%, that's 23 of them, that's 76% also mentioned regarding the long-term effects of their medicines. 33% disagreed, stating that their medicines are a mystery to them. And 53% disagreed that the medicines disrupted their life. 33% had a worry, disagreed about worrying that they would be dependent on their medications. However, 30% of them did mention that they are worried about being dependent on the medicines. With general overuse and harm, 40% of them disagreed that the doctors use too many medicines, but 23% agreed on this. With 76% being uncertain that natural remedies are safer than the medicines, 13% of them agreed to it. With doctors placing too much of trust on medicines, 73% were uncertain to answer, with 13% disagreeing on it. With 83% being uncertain whether the doctor had more time to spend, they would be prescribing fewer medications, as they said that the patient, as they grow old, they have more comorbid conditions. So even if the doctor gets more time, they may not be able to reduce the number of drugs being prescribed for their different comorbid conditions. Sorry. So in the general question regarding the harm from the medicines, 70% of them disagree that the people who take their medicines should stop taking their medicines for a while every now and then. And for most of the medicines are addictive. To this, 46% disagree. However, 30% of them agree. Medicines do more harm than good. 53% disagreed on this, and 43% of them were uncertain to answer. All medicines are poisons. 53% of them disagreed, and 46% of them were uncertain to answer this question. Coming to this chi-square test that was conducted to find the relationship between the gender, literacy, number of comorbidities, area of domicile, and the socioeconomic status, gender was found to be significantly associated with the medication adherence with a p-value less than 0.05. However, there was no statistical significant difference found between the two groups regarding their beliefs about the medicines. To conclude, the belief about medicines plays a role in the medication adherence behavior. However, in the current study that was conducted for a short period of time with a small sample size, was the limitation to find any significant findings. A clinical pharmacist can play a vital role in identifying the medication adherence behavior through such surrogate markers and provide the necessary counseling, thereby promoting the medication adherence. Thank you. Hello.
Nice presentation, Jihad.
Thank you, sir.
If any question, anybody can proceed, please.
Okay, I have a few queries.
Yes, sir.
It was a nice study that you have done a good study on the belief about medication. Have you taken a permission from the developer of the BMQ questionnaire or standardize it, or how?
So we have taken the permission, sir.
Okay.
And
so,
actually, this is just like a pilot study kind of a thing. We would be working on like further to increase the number of patients. So to understand significantly how that plays a role.
okay. And for the Indian context, have you modified the questionnaire, or you are using the same similar?
No, sir, we have not modified, because if we have to modify, then again I have to go through the validation of that questionnaire and conduct the study then.
Okay, definitely. Yeah, thank you.
Yes, sir.
Similar with the SMQ, have you found the correlation with this and belief and the adherence?
Yes, sir. We found like the two groups, the adherent group and the non-adherent group. We checked for their belief about medications. They like how many % of them agreed. The agreed percentage we have taken and seen, but there was no significant difference found between both the groups. So we assume that because the sample size was small, we did not get any significant values.
How many number of patients you have used? Only 50 or something?
30 totally, but then 15, like 50% medication adherence was good, and 15 of them was not.
Okay. Okay. Thank you.
Thank you, sir.
Any other question? Okay. Okay. Thank you very much, Ms. Jihad Sayed.
Thank you, ma'am.
Okay, we have the next presentee, Mrs. Sudala Shirisha. Are you here?
Yes, ma'am.
Ms. Sudala, okay. Mrs. Sudala Shirisha is a PhD scholar at University Department of Pharmaceutical Sciences, Utkal University, Bhubaneswar, Odisha, and she will be presenting on the topic Nisoldipine loaded solid lipid nanoparticles and nanostructural lipid carriers design and evaluation. Mrs. Sudala Shirisha, you can now present.
Yes, ma'am, my screen is visible now.
Yes, but your voice is very low.
Oh. Now it is clear, ma'am.
No, it's not clear.
No, still
If
you
you are
can.
using your mobile phone, please use the landscape mode.
Okay, sir.
Kindly tilt it and then we'll see it big.
Yes, sir.
Little better.
Yeah.
A very good afternoon to one and all. Myself, Sudala Sirisha. I am a research scholar from the UDPS Utkal University, Odisha. Today I am going to discussing about the topic is nisodipine loaded solid lipid nanoparticles and nanostructure lipid carriers design and evaluation. So these are the contents I am going to discussing: introduction, methods, materials, result, discussion, conclusion, and references. So coming to first introduction part. So nisodipine, it is one of the antihypertensive drug under the class of calcium channel blocker. Under the chemical category, it is a dihydropyridines. And the main aspect here, why we are selecting the nisodipine drug, due to the low bioavailability, that is only 5% when we are taking the oral administration, due to the first pass hepatic metabolism. So that's why we are going to formulating into the transdermal form. So before going to transdermal form, we are going to incorporating into the SLN and NLCS particles, then incorporating into the gel form, that is a transdermal hydrogels. So based on the physical and chemical properties of the nisodipine, it is a suitable drug candidate for the formulating into the transdermal drug delivery. And why we are selecting the particles here, that is a vesicle system. So vesicle system is also one of the enhancement of the permeation technique for the transdermal delivery. Why? Because during the transdermal delivery, we have a statin corneal layer for the inhibiting, for the preventing the permeation of the most of the drugs. So for overcoming that disadvantages, we are formulating into the vesicle form. So vesicle, nothing but we are using the lipid molecules. So those lipid molecules can enhancing the permeation across the statin corneum layer. So next, coming to the materials and methods. The first one is characterization by the differential scanning colorimetry, that is the DSC studies. This is one of the drug and excipient compatibility studies. So here we are using the DSC studies. So in that we are determining the or quantifies the enthalpic changes during the endothermic and exothermic effects. So here we are using the 5 mg sample, and also the heating rate is at 20 degrees centigrade per minute, and also heating range is 20 to 200 degrees centigrade. And next, coming to preparation method, after confirming there is no compatibility, there is no interaction between the drug and excipient, then only we go for the formulation. So whatever the result we are discussing in a later. So coming to first one is the preparation method. So preparation method. So first we are taking the drug, and that can be dissolved in a solid lipid. So here we are taking the lipid is a dinosine, that is a trimyristic acid. Dinosine 114, 118, 116, and also the surfactant, co-surfactant. So first we are preparing the oily preparation. Oily preparation, nothing but that is a lipid portion. And that lipid portion, first we are taking into the solvent mixture, that is a chloroform and methanol 1:1 ratio. And after that, by using the rota evaporator, we had to evaporate the whatever the solvent. So we are getting the single layer, that is a layer of the lipid layer. The same time, simultaneously we are preparing the aqueous preparation. So in that we are taking the another surfactant. So these two, whatever that oily portion and the aqueous portion, the two portions we had to maintain the same temperature. And after that, the aqueous portion we had to add it to the oily portion. We are getting the first one is the emulsion form. First, we are getting the emulsion form, that is a water in oil type of emulsion. So after homogenization and ultrasonication process, we are getting the solid lipid nanoparticles, and also for the preparation of nanolipid structures. For that purpose, whatever that solid lipid we are replacing, some amount of the solid lipid we are replacing with the liquid lipids. Here we are using the oleic acid. 30% of the solid lipid we are replacing with the oleic acid. We are preparing the nanolipid structures. And the next one, this is a coming to formulation table. See here in a formulation ingredients, we have a nisodipine. In all the formulation, we are taking the 10 mg. And also we have a dinosine 114, 116, 118. These three are solid lipids. These are the solid lipids. So for the optimization of the amount of lipid, we are taking the different concentration. That is the different amounts we are taking. That is 100, 200, 300. So in that we are based on the different evaluation parameters like a particle size, zeta potential, and drug content, entrapment efficiency, in vitro drug release. Based on all the properties, we are selecting the F2, that is a 200 mg. And after that, solid lipid, after that soya lecithin and poloxamide. These are the surfactant co-surfactants. So these can be also we had to optimize it. So for that purpose, we are taking the different concentration and then we are conducting all the parameters. Based on all the evaluation parameter, we are optimizing the formulation ingredient amount. And the oleic acid. See here, the oleic acid is the last one, that is the NLC, that is a nanolipid structures. So in that, solid lipid can be replaced with the oleic acid. And the distilled water is the solvent, and the chloroform methanol is also solvent mixture. That is 1:1 ratio for the preparation of the oil portion. So after getting the solid lipid nanoparticles and also the nanolipid structures, that can be incorporating into the gel. So here for the gel formation purpose, we are taking the gel forming layer, gel forming polymer, that is we are taking the carbopol 934P. So here for the preparation of the gel also we are using the homogenization process. And next, so before preparation, before incorporating into the gel, so first we had to evaluate the nanoparticles. So for that purpose, the main parameter it is particle size, polydispersibility index, and the zeta potential. So that can be, we can estimated by the photon correlation spectroscopy, that is a Malvin zeta sizer we are taking. So based on that, we are determining the size and the PDI and the zeta potential. And next, determination of entrapment efficiency by using here, we are using the Centrisat ultracentrifuge tubes. So that can be used for the entrapment efficiency. And here for the entrapment efficiency, first we are determining the unentrapped drug in the aqueous portion. Unentrapped drug in aqueous portion by using the ultracentrifuge tubes, and based on that amount we had to include it in this below equation, that is entrapment efficiency equal to amount of nisodipine in lipid phase. So we are getting the unentrapped, that is aqueous phase, the total drug content minus unentrapped, we are getting the drug which is entrapped in a lipid portion by the SA value. So that is a drug content value into the 100. So for the determination of total drug content based on the dilution process, just we are taking the sample that can be diluted by using the solvent mixture, that is the chloroform and as well as the methanol. And also the further dilution we are required. Then the last sample we are injecting into the UV spectroscopy and we are determining the drug which is present in a sample at the lambda max 238 nanometers. And here the in vitro release studies by using the dialysis bag method. So in a dialysis bag method, what we are taking the 100 ml beaker, and in that we are using the boiling tube, and the bottom of the boiling tube we had to tie with the dialysis membrane. The dialysis membrane that can be soaked in a water overnight period. So and after that can be tied into the boiling tube ending point. So within the boiling tube we had to place the sample, that is a gel, otherwise SLN and NLCS particle solution, whatever it is. In the 100 ml beaker we are filled with the 75 ml of buffer, that is a phosphate buffers, that is 7.4 pH. And also we had to add the SLS for maintaining the sink condition, and also that can be placed in a magnetic stirrer at the RPM 12,000 to 14,000 meter RPM. And ex vivo permeation studies, that can be used by the French diffusion cells. So here the same French diffusion cells we are using in a donor compartment. We are placing the SLN and NLCS portion, and the receptor compartment we are placing the buffer, and also in that also we are taking the 0.75% of SLS for maintaining the sink conditions. And after that, we are conducting the physical stability study and also the SLN and NSCS, that is the nanoparticles. So compulsory they are having the stability problems. To overcoming this stability problem, we go for the lyophilization by using the cryoprotecting, that is a 10% mannitol we are using. So coming to result and discussion part, it is a differential scanning polarimetry. In that we are using comparing between the nisodipine pure drug and comparing with the all the other formulation, that is a dinosine 114 plus nisodipine, dinosine 116 plus nisodipine, dinosine 118 plus nisodipine, and every thermogram there is a showing the peak of the pure drug, but the intensity will be decreases. Why the intensity will be decreases here? The drug it is incorporated into the solid matrix. The lipid matrix can be surrounded by the drug molecule, so that's why the intensity will be decreases, but the peak is present. So that's why there is a no interaction, only there is a entrapment. It is not a chemical interaction, it is only physical. So based on these studies, there is no drug and excipient interaction is there. And coming to the optimization of formulation and the process variables. So process variables and the formulation variables here, the formulation variables to know the optimum concentration of the lipid and the surfactants, co-surfactant. And here the lipid concentration, when we are increasing the concentration from the 100 to 400, here what happened, that can be affecting the particle size of the nanoparticles, SLN and NSCS in both cases. So that's why we are optimizing the concentration at the 200. Why? Because their range is our range, that is the 170 nanometers to 200 nanometers. So when we are increasing the lipid concentration, what happened, then again increasing the particle size also. So that's why we are taking the optimum concentration of lipid is a 200 mg. And similarly, soya lecithin also we are taking the 50, 100, 150. Here also, when we are increasing the concentration, there is also increasing the particles, that is a 190 to 230. So in that cases, we are taking the 100 mg is a soya lecithin, and also the poloxamer is also we are taking the 1.75. So based on the particle size range, we are selecting the concentration of the different formulation ingredients. And similarly, the process variables. The process variables here, we are taking the homogenization time. That is, first we are starts with the 4 minutes and the 8 minutes, 10 minutes, and the 15 minutes. In the 4 minutes, there is a particle size above 400 nanometers, and the 8 somewhat decreases, and the 10 it is also decreases. But at the 15 minutes, what happened? There is a further there is no changes in the particle size. So that's why we stopped at the 10 minutes. And similarly, ultrasonication. Ultrasonication, we are using the ultrasonicator, that is a probe sonicator, that is a 12T probe sonicator we are using. So we are taking the 15 minutes, 20 minutes, 25 minutes. Here also the same thing we are observing, that is a 20 minutes we are taking as the optimum time for the ultrasonication time. So based on these factors, we are selecting the process variables and as well as the formulation variables. And after that, when we are preparing the vesicle, that is a particle system, compulsory entrapment efficiency is also important. Why? Because this entrapment efficiency can be affecting the permeation studies. So when they are having the high drug loading capacity, they are showing the sustained action, that is extended release of drug from the vesicle. So that's why entrapment efficiency is also very important. Here we are using the centrisort tubes. So here the two chambers are there, that is outer chamber and the aqueous chamber is there. So in outer chamber, we are placing the sample, and the aqueous chamber, that aqueous chamber ending, we are placing the membrane. We are placing the filter membrane, and this total tube can be ultrasonicated. So during that ultrasonication, what happened? So whatever the based on the density that can be separated. So the aqueous portion can be moving towards the upward. Yes, sir. Yes, ma'am.
So can you try to complete it because the time is running out?
Okay, sir. Okay, sir. So based on that, the aqueous portion can be separated. From that aqueous portion, you had to calculating the unentrapped. From that unentrapped, we are following the formula to find out the entrapment of the drug. So these are the physicochemical parameters, that is the size, PDI, zeta potential. So based on the concentration of the lipid, the effect is based on the size, PDA is effect, PDS, and also zeta potential is. So these are the graphs. See here from the F1 to F5, that is the yellow portion. So in F3, so that is showing the high PDI, sorry, high particle size. Similarly, so there is a up and down, but we are taking the optimum concentration is a 200. And this is the second, that is the figure 4 is also showing the formulation port, entrapment efficiency, and as well as the drug content. This is the transmission electron microscopy. This is a give the information support to the particle size. So whatever that particle size we are taking from the photon correlation spectroscopy, it is also nearer to the transmission electron microscopy, that is a 150 to 300 nanometers. So this is a important test for the dermal delivery, that is occlusion test. Why? Because which having the high occlusion capacity, they are going to form as a one smooth layer on the surfaces, and also they are having the highest retention time on the skin surfaces. So for that purpose, we are using this occlusion test. And here the SLN and NLCS compared to the NLCS, SLN showing the highest occlusion capacity. And this is an in vitro drug release. So all the formulation, the E4 NLCS, they are showing the 82% in 24 hours time. And when we are incorporating into the gel, the E4 NLCS, they are showing the highest, that is only 40%. Why? Because they are incorporating into the gel, so that's why they are delaying the drug release. So based on that kinetic parameters, that is we are fitting into the parameter, that is order of release and also the pattern of drug release. So here all the formulation, they are following the first order kinetics, and also they are following the Iguchi, that is the diffusion model of the drug release. And here the XY, so based on just the conditions are different, but here also the same result we are getting, that is E4 NLCS and the E4 NLCS gel, they are showing the highest drug permeation. So based on the XY data, we are determining the flux. So in the all the flux, it is in case of E4 and E4 NLCS, they are nearer to the drug solution flux. And this is the important one, that is there is a correlation between the in vitro data and ex vivo permeation. So in a different two conditions, but where we are having the good correlation between the in vitro data and also the ex vivo data, they are nearer to the 0.95 to 0.98 in all the formulation, not single formulation. In all the formulation, we are showing the good correlation. So this is for the SLN and NLCS particle, and this graph shows the for the gel formulation. Here also they are showing the good correlation. And the skin irritation studies, here we are using the control in SLN and NLCS formalin. Formalin, it is a reference control without any formulation. And one group we are placing the SLN and another group we are placing the NLCS. At last, after working over 7 days, there is no showing in edema, erythema in all the rats. And coming to these are the stability conditions, stability studies. Here we are placing at the room temperature and at the refrigerator conditions, and we had to testing the sample at every 30 days and 60 days. At the first day we are testing, and after 30 days and after 60 days, there is a slight increasing in the size and also polydispersibility index and zeta potential. Why? Because during the storage, what happened, there is aggregation of the particles, so that's why there is a slight changing in the parameters. So after that, so compulsory we have to overcoming the stability problem. We are going to the lyophilization. Coming to conclusion, so based on the our studies, so the SLN and NLCS is also one of the good enhancement technique for the dermal delivery. So in case of antihypertensive, not only antihypertensive, which drugs are undergo the extensive first pass metabolism, so compulsory we had to formulating into the dermal delivery. It is a very good approach. So we are further to study for the in vivo conditions also.
So by using the rabbits. And these are the references. And finally, I'm thankful to my academic guide. It is a Yamsani Malsudan Rao Garu, and also my mentor Sunit Kumar Sahu, and also I'm thankful to the financial support from the Women Scientist A, Department of Science and Technology, Government of India. And also I'm thankful to the UDPS Utkal University, Odisha, for giving the facility for doing my work. Thank you, sir.
Thank you very much, Ms. Sudala Ashisha. It was a very nice and elaborative presentation. And I welcome if any chairperson have any questions. Anybody, any question?
We'll proceed, ma'am. Still any member? How many members left, ma'am?
One member. One last presentation is remaining.
Okay.
Okay. Again, Mrs. Dala, thank you for your participation, and we wish you all the best.
Thank you, ma'am. Thank you. Thanks so much.
And now we proceed for the last presentee, Ms. Samaksha PB. Ms. Samaksha, are you here?
Yes, ma'am.
Okay. Ms. Samaksha PB is a research scholar at Department of Pharmacy Practice, JSS College of Pharmacy, Mysuru, Karnataka. And she will be presenting on the topic medication therapy management services in patients with psychiatric illness, a collaborative study. Ms. Samaksha, you can please present now.
Yes, ma'am. Ma'am, can you just enable my screen sharing? I'm unable to share my screen. Yes. Namaste, everyone. My topic is on medication therapy management services in psychiatry, patients with psychiatric illness, a collaborative study. So we know that medication therapy management services is to optimize and identifying, resolving the drug-related problem and resolving the queries. We know that the clinical pharmacist has evolved from the dispensing to the community services also. So medication therapy management is a part of the service providing to the patient for the patient better care. So this study has optimized the medication therapy management services in the psychiatric patients to obtain the clinical outcome, good clinical outcome, and to resolve the drug-related problem, and also to resolve the complaints on the medication non-adherence. So coming to the methodology, the ethical clearance was taken from the Institutional Ethics Committee, JSS Medical College and Hospital, Mysuru, and the study site was opted. It is a joint initiative by the Department of Psychiatry and the Department of the Clinical Pharmacy. And also the study design is a cross-sectional study. Duration was the study of June 2021 to December 2021. So when the patient visited the psychiatry OPD, the clinical pharmacist reviewed all the case sheet and assessed the inclusion criteria and exclusion criteria. The patient who are aged 18 years and above of any gender and diagnosed of depression, bipolar affective disorder, and alcohol dependence syndrome, or any psychiatric illness of these four, and who gave the consent were enrolled in the study. The patient who had multiple comorbidities, psychiatric comorbidities, such as a bipolar affective disorder with the alcohol dependence syndrome, or a schizophrenia with alcohol dependence syndrome, all these patients were excluded from that study, and the patient who had cognitive impairment were also excluded. Patients who met this study criteria were assessed drug treatment chart, and also if any drug related problems were identified, we have implemented the strategy to resolve the drug related problem, and also we have given the solution for the non-compliance of the medication non-adherence. And all after the medication therapy management services, we have assessed the clinical outcome and documented the document from the baseline to the we have conducted 2 follow-ups, like a follow-up 1 and a follow-up 2. Follow-up 1 and follow-up 2. So after that, we have followed each patient over the telephonically for the reappointment with the psychiatrist also. So coming to the results and discussion, so we have enrolled almost nearly 189 patients who met the study criteria under the age of 18 to 30 years, and majority of the patients were female, that is 55%, and most of the patients had a normal body mass index. 74% of the patients were illiterate, and 76% of the patients were unemployed, and overall 33% of the patient income was less than 3,000. And coming to socioeconomic status, as we know, 32% of the overall population had a lower socioeconomic status with a rural background of 60%. Among the 4 psychiatric disorders, depression was a majorly seen diagnosed patient, that is almost 46%, with a medical comorbidity of 7%. Coming to the management therapy services which we have given to the patient, so 189 patient counseling was provided to the patients, followed by pharmacist intervention, that is 126 36% of the interventions were provided, and 10% of the drug informations were provided. So drug information, we can see like ADR was a question category which as asked by the postgraduates as well as the consultant, and followed by drug interaction, dose or dosage administration or drug therapy indication, pregnancy category, and lactation, whether the medication can be given or not, and the efficacy of the medication, mechanism of action. So most of the queries were related for the patient better care, that is 77%. And the drug information inquirer most of them were postgraduate medical students, followed by consultant and interns. So majority of the time, the access for mode of request was a direct access during the OPD basis. And interventions were provided almost, as I said, 126 interventions were provided. So among which adverse drug reactions were seen in depression patient, that is 19, and also subtherapeutic dose was commonly provided intervention in the psychiatric patients. For example, due to financial issues or due to the non-availability of the psychiatrist, they could not meet the psychiatrist, so they were breaking the tablet and they were consuming the medication. And drug-drug interaction, for example, patient who had was known case of hypothyroidism receiving thyroxine, and also patient who had similar gastric complaints was given with pantoprazole. The drug interaction with pantoprazole and thyroxine. Overdose was like, for example, I'm telling one patient was prescribed with 0.25 mg of clonazepam, and patient already had the medication, so the dose was increased to 0.5. So psychiatrist, because to decrease the economic burden, asked the patient to take the two medication of 0.25 mg. But since the patient could not assess what the psychiatrist was saying, for a couple of days, the patient was receiving 2 tablets of 0.5 mg. That was overdose of clonazepam. And improper drug selection was seen only in the alcohol dependence syndrome patient. So the thing was like the drug use without indication was seen in depression, bipolar affective disorder, as well as schizophrenia. So we saw as a patient did not had any complaints of nausea or vomiting, but prescribed was with the pantoprazole plus domperidone combination. And untreated indication was seen in depression as well as the alcohol dependence syndrome for constipation and diarrhea, dry mouth. So majority of the intervention which we provided, which clinical pharmacists Sir? Okay, sorry. Majority of the intervention which we provided were accepted, and there was a change in the therapy, and only 6 of the interventions were not accepted, and that was a minor and major intervention. So coming to the patient counseling, when we are analyzing for the enrollment at the baseline, we educate the patient regarding the medication use, how to take the medication, when to take the medication, and some of the precautions to take before the medications, like need dry mouth is there, we'll ask the patient to consume the watery content of fruit like cucumber or watermelon, such kind of thing. And also we assessed the medication adherence. So we came to know that majority of the patients were not adherent to medication at the baseline, that is 61%, among which depression were like a 62 patient who diagnosed with a depression were not adherent to medication, followed by BPAD 34. So the major barrier for the non-adherence was patient related, followed by provider related and also drug related. So when we assessed individually what are the barriers, we came to know that because of a lack of knowledge, about the disease and the medication, there was a decrease in the non-adherence, followed by financial issue, forgetfulness, self-assessing. The patient will take for 15 days and then until symptoms were subsided, and they'll stop the medication, self-assessing, and myths about the psychiatric medication and psychiatric disorder, busy work schedule, lack of support from the family and the friends, and followed by drug-related problems due to the ADR, due to the non-availability of the medication, and also due to the non-availability of the pharmacy. So provider related, when there was a non-availability of the psychiatrist, the patient was not adherent to medication. So we created a strategy to resolve all these barriers, where we reduce the medication adherence from the baseline to follow up to 20%, baseline it was 61 and now it is 46%. So we took the individual barrier and we started providing the services on the individual basis. Like a poor medication, we counsel the patient regarding the disease, and financial issues, we collected the number from the NGOs and also the pharmacies, where they are providing 10% or 30% discount for the total medications. We contacted them and we gave the patient the pharmacy number, and we asked the patient to go there. And also free medications were available in our hospital. Free medications are in the sense like when the patient get discharged from the inpatient, we'll ask the patient caretaker whether are they going to take back the leftover medications. If they donate to the hospital, we have a separate section for that. So according to the patient prescription, we will provide the free medication to the patient. Forgetfulness, when the patient complain of forgetfulness, we'll assess whether is it because of the drug, or is because of the age related, or is it because of some other underlying cause. So we'll refer to the neurology so that if any early onset dementia is there, or is it because of a dementia. And myth and misconception, we'll sit with the psychologist and we'll counsel the patient regarding the and we'll try to erase the myth and misconception what they have on psychiatric illness. And also lack of family support. We'll have a session with the patient and patient caretaker. We'll ask the patient caretaker to support the patient, and will try to convince the caretaker to support the patients regarding that. And busy work schedule, will ask the patient to schedule their activities. And also we have asked a few of the patient to download some app so that it won't affect their busy schedule. And drug related problem, adverse drug reaction was there, and if it was mild adverse drug reactions, we have counseled the patient, and if it is moderate, we have de-challenged the medication and changed the other class of a drug. Non-availability of the medications, we have soon after the prescription they get from the psychiatrist, we'll call the pharmacy and we'll ask the pharmacist whether these medications are available available or not. So before they go into the random pharmacy, we will direct the patient to the pharmacy where all the medications are available. So, and provider related unavailability of the psychiatrist, we call the patient on the, if the follow-up is for 1 month, we have called the patient on 28th day. We'll say the psychiatrist will be, your consultant will be available on this day, please come by. We'll give a appointment kind of thing, reminder call. And also difficulty in assessing pharmacy, as I told you, that will ask the patient to go the nearby pharmacy. If they are unable to go nearby the hospital, we'll ask them to go near their residency. So same as usual, after providing these MTM services, the medication non-adherence rate has drastically decreased to 11%, from 61 to 11%. The same strategy was applied to all the barriers the patient provided for non-adherent to medication. So coming to the follow-up, you can see that the medication adherence rate has been increased over the non-adherence. So finally, I summarize that this study has proven that the need of clinical pharmacy as in a multidisciplinary care will definitely improve the health outcomes and for a better patient quality of life. Thank you.
Thank you very much, Ms. Samaksha.
Thank you, ma'am.
Any question?
Yeah, thank you very much, Ms. Samaksha. It's very comprehensive and good presentation from you.
Thank
and
you,
then
sir.
the work is very nice as well.
Thank
And
you,
definitely there is a role of clinical pharmacy in the good intervention for the patient to establish a GCP.
Yes, sir.
My question is this.
Yes, sir.
When have you calculate the burden on the patient when you are having a ADR related issues? Okay. In your
Yes,
study.
sir. Yes, sir. We did not assess in this study, but we have come across the burden, sir. Actually, so, when the patient had this medication non-adherent, because of the ADR, they'll stop the medication, then they won't take the medication. Because of that, the relapse has happened and the patient got admitted. Because of the relapse and because of the hospital stay, the economical burden has increased the patient. That's why we are constantly counseling the patient regarding the importance of a medication adherence. For each follow-up, even though we have already explained them the importance, but for each follow-up, we'll counsel the patient as well as the patient caretaker. Even the patient is self-managed by the medication, we'll ask the caretaker to cross-verify whether the patient is taking the medication by pill count method. So that's how we came up with the strategy to reduce the burden.
Okay, thank you very much. Very good presentation.
Thank you, sir.
Thank you again and congratulations to all the participants. And I would like to thank all the chairpersons for your time and active participation in judging these sessions. And with this, we conclude this session. And we will move.
Judges, Dr. Mittal.
Yes, sir. I'm sending you the
Dr. Gupta, please forward me the results.
Yes, sir.
And we, as soon as I will compile, we will start the award announcement section.
Sure, sir.
So we are compiling the result of oration competition. So just 5 more minutes are needed. Thank you. So I welcome all of you in the award ceremony of the first day of APP 15th Indo-Malaysian Conference. So I will share the PowerPoint presentation and we will flow accordingly. So are you able to see my screen? Anybody can confirm?
Now it is not visible, sir.
No, sir.
Screen is not visible?
No, sir.
They're not visible.
Now?
Yes.
Yes, sir.
Yes.
Now?
Yes.
Yes, sir. Yes, sir.
So I will move the slide so and you can say yes. That's
Yes, sir.
things are moving?
Yes, yes. Yes, sir.
If it is stopping in between, you can let me know in between. Okay.
Sure, sir.
Okay, so I welcome all of you in the virtual award ceremony of APP 15th Indo-Malaysian International Conference. So we have few announcements. So slide got changed?
Yes, sir.
Okay. So today we are announcing our new local branch, which is APP Haldia Local Branch 2022. It will be led by Professor Suman Patnaik and Professor Lakshmi Kant Kathal from Haldia Institute of Pharmacy, Haldia, West Bengal. So congratulations, Professor Suman and Professor Lakshmi. Hope to see branch growing well under your leadership. If you want to share, Professor Suman, if you want to share your views, you can.
Okay, good afternoon, everybody. So first of all, I thanks to Honorable President Dr. Rajiv Dahiya sir and Dr. Sunita Dahiya sir, and a special thanks to my friend Somnath De to advise me to be a member of the APP. Then I approached that I will be give my best efforts to fulfill the goals and objectives of the society of this organization, and I encourage all pharmacy professionals to be the member of this organization and ensure the organizational growth. Okay, thank you, sir. Thank you for giving me this opportunity.
Okay. So congratulations, Professor Suman and Professor Lakshmi. So we'll move next.
Thank you.
So we have upgraded our APP Biotech division. Dr. Paramita Basu will be our new international head of the division, and Professor T.S. Shanmuga Rajan will be the new national head of the division. And Dr. Basu is associate professor at Toro College of Pharmacy, New York, and Professor Shanmuga Sundaram is a professor of biotechnology at School of Pharmaceutical Sciences, Vistas, Pallavaram, Chennai. If Professor Shanmuga Rajan is there, he can share his views.
Yes, sir. Respected Dr. Rajiv Dahiya, Founder, President, APP Scientific Committee Chairman, Director, School of Pharmacy, University of West Indies, and respected Dr. Sunita Dahiya, Organization Chairman, General Secretary, APP School of Pharmacy, University of Quito, USA, and respected Dr. Madhu Gupta, Scientific Committee Co-Chairman, Delhi Pharmaceutical Sciences and Research University, respected Professor K. Padmalata, Convener and Principal, Vijaya Institute of Pharmaceutical Sciences, and Dr. Rajita Gala, Co-Convener and Vice President, APP, for introducing me as one of a national chairperson for the biotechnology division. And thank you very much for selecting me and also introducing this 15th Indo-Malaysian Conference. It's clearly one of the most significant events of my professional career. This recognition is another example of the great support that you have given us over the past. I'm very fortunate to affiliate with the Association of Pharmacy Professionals. I know still I much to learn, but with the continuous support and encouragement from people like you. Thank you very much, sir.
Thank you. So we will move next regarding awards. So first in the international category, 2 women are selected. So first award is Young Pharmacy Teacher Award, and it goes to Dr. Sivkanya Floria, who is Associate Professor at Department of Pharmaceutical Chemistry, Faculty of Pharmacy, AIMST University, Malaysia. So congratulations, Dr. Floria, for Young Pharmacy Teacher Award 2022. if you are with us, you can share your views. Okay, we can move next. The next award in international category is Young Investigator Award 2022, and which goes to Dr. Jisa Lusa, who is Assistant Professor of Pharmacy Practice at College of Clinical Pharmacy, Imam Abdul Rahman bin Faisal University, Dhamam, KSA. Dr. Lusa, if you are with us, you can share your views.
Okay. Yes, thank you, Doctor. Good afternoon, everyone, and thank you for considering me for the research award. And I would like to introduce myself as a clinical pharmacist rather than a researcher in the neuropsychiatry department. Also, I'm an academician as well as a researcher. My research is mainly focused on the pharmacy practice areas that included but not limited to the healthcare outcomes, drug optimization, drug safety monitoring behaviors, and social aspects of the pharmacy practices. And so I have almost 50 publications, as you can see in my biodata. And thank you for once again considering me for the APP award.
Thank you, Dr. Lusa.
Thank you, Doctor.
So we will move to our next category. It is national category, and we have selected one candidate in national category, Young Talent Award 2022, and this award goes to Dr. Ritu Kataria, who is principal at GVM College of Pharmacy, Sonipat, Haryana. Dr. Kataria, if you are with us, you can share your views. Okay, we can move next. Regional and outer state category. So we have selected 7 to 8 students from different states as in best student, along with one outstanding research scholar award. First award is Outstanding Scholar Award from Odisha State, and the award goes to Ms. Sudala Sirisa, who is Research Scholar at Department of Pharmaceutical Sciences, Utkal University, Bhubaneswar, Odisha. Congratulations, Ms. Sirisa. If you are with us, you can share your views. Okay, we will move next. In Best Student category from Rajasthan State, the award goes to Ms. Komal Chobisa, who is B.Pharm second year student of US Ostwal Institute of Pharmacy, Mangalwad, Chittorgarh, Rajasthan. Ms. Komal, if you are with us, you can share your views.
Good evening, everyone to present here. Myself, Komal Chobisa, student of B Pharmacy 4th semester, US Ostwal Institute of Pharmacy, Mangalwad. Present here to thank you for granting me such an honor and a special award. I feel overwhelmed and emotional, and I am lost of words. This is very special moment in life. The dedication of my award, I would like to extend to my parents and the teachers of US Ostwal Institute of Pharmacy. What I am today is because of my parents and teacher. Furthermore, my parents left no stones unturned in providing me such the resources or in my life. Most noteworthy, I can never pay back what they did for me. Thank you for giving me such an opportunity. Jai Hind.
Thank you, Ms. Komal. From Delhi State, the Best Student Award goes to Ms. Jyoti Kumari, who is M.Pharm second year student at Department of Pharmacology, DIPSAR, DPSRU, New Delhi. Ms. Jyoti, if you are with us, you can share your views. Ms. Jyoti?
Yes, good afternoon, everyone presented here.
You can
I'm from.
share your views.
Am I
Yes,
audible?
please. Yes, please. Yes, you are audible.
Good afternoon, everyone presented here. Actually, I am from Nepal, but currently studying in Delhi Institute of Pharmaceutical Science and Research in Delhi. And I have completed my bachelor from MMU in Ambala, Mulana. And first of all, thank you for honoring me through this award. I am feeling very happy to join APP Association membership. And today it's been a very fruitful and informative session for me. That's all. Thank you, sir.
Thank you, Ms. Jyoti. From Uttar Pradesh State, the Best Student Award goes to Ms. Ria Kaushik, who is M.Pharm student at Department of Pharmacology, ITS College of Pharmacy, Muradnagar, Ghaziabad, Uttar Pradesh. Ms. Ria, congratulations. If you are with us, you can share your views.
Good afternoon to everyone. Thank you for such a great award. And I'm very happy and glad to attend this conference. And this is like a wonderful conference I have attended. I am from AKTU College. I am from ITS College, affiliated by AKTU University in Lucknow. And I have learned lots of things in this college. I have also completed my B Pharma from the same college, and I have learned reviewing articles, learned so many subjects which are needed in pharmacy field. And I'm glad to have this award. Thank you for the opportunity.
Yeah, congratulations, Ms. Ria. From Karnataka State, the award goes to Ms. Samaksa PB, who is research scholar at Department of Pharmacy Practice at JSS College of Pharmacy, JSS Aher, Mysore, Karnataka. Congratulations, Ms. Samaksa. If you are with us, you can share your views.
Thank you. Namaste, everyone. I thank the organizers, research committee, and scientific committee for enrolling me into this. I thank each and everyone. I thank my guide, Dr. M. Ramesh, for giving me such platform to and encouraging me in all my innovative works in area of psychiatric pharmacy. I hope this organization will much more encourage the female researchers like us. Thank you very much, everyone. Namaste.
Congratulations, Ms. Samaksa. From West Bengal State, the award goes to Ms. Dipanvita Bera, who is B.Pharm student at Haldia Institute of Pharmacy, Haldia, West Bengal. Congratulations, Ms. Bera. If you are with us, you can share your views.
Good afternoon to everyone present here. I am Dipanita Bera, B.Pharm final year student of Haldia Institute of Pharmacy, and I am very glad to deliver my views on such a momentous occasion in my life. It is a huge honor for me to receive the Best Academic Award for being a student of Haldia Institute of Pharmacy. I am grateful to Haldia Institute of Pharmacy for providing all facilities to study and build my career. I would like to thank our principal sir, Professor Dr. Suman Patnaik, and Vice Principal Sir, Professor Dr. Lakshmikanto Kanthal, for selecting me this award from my institution. I am deeply grateful to our college management, Principal Sir, Vice Principal Sir, and all faculty members for continuous support and guidance on my B.Pharm career. I express my gratitude thanks to the all respected members of Association of Pharmacy Professionals and Vijaya Institute of Pharmaceutical Sciences for Women for this recognition of my academic score. I would like to dedicate this award to the institution. The dedication of this award I would like to extend to my parents. I am also thankful to my all classmates in our college. This award will motivate me to achieve more success in my future. Thank you.
Congratulations, Ms. Dipanvita. We have Vice Principal of the college with us. So if he wants to share his views regarding the branch as well as about Ms. Dipanvita.
స్టూడెంట్ అవార్డ్స్ ఏవి ఇస్తున్నారు మేడం. బెస్ట్ స్టూడెంట్ అవార్డు Haldia Institute of Pharmacy, West Bengal.
The vice principal of
Lakshmikant, you can share your views.
International Women's Day కి ఇస్తున్నారండి. అది ఇది రెండు కలిపి ఉన్నాయి సర్టిఫికేట్ లో.
Please unmute.
Yes, sir. Good afternoon, one and all. So, myself, Dr. Lakshmikant Kansal, Vice Principal of Haldia Institute of Pharmacy, and we are grateful to Dr. Rajiv Dhaya sir, Honorable President of APP, and also Dr. Sunita Dhaya, Madam, Secretary of APP, and entire team of APP for giving our student the opportunity to get the award. And thank you, all of you.
Thank you, Dr. Lakshmikanta. And I hope the branch, local branch will grow under your able leadership in the West Bengal.
Thank you, sir.
Thank you. So congratulations, Ms. Dipanvita. From Odisha State, the award goes to Ms. Akanksha Sahu, who is BPharm second year student at Department of Pharmaceutical Sciences, Utkal University, Bhubaneswar. Congratulations, Ms. Sahu. If you are with us, you can share your views. Ms. Akanksha?
Yes, sir. Thank you, sir. Thank you, sir. Very good evening to one and all present here. And myself, I'm Akanksha Sahu. I'm a student of BPharm 4th year, University Department of Pharmaceutical Science, Utkal University, Bhubaneswar. And it's a very great honor for me to be a part of APP family, and I will feel very privileged to have attended today's conference. And I would really thank my professor of University Department of Pharmaceutical Science, and a special thank to Sunit Kumar Sahu, sir, for guiding me and for recommending me for this award. And I feel very overwhelmed. And thank you, sir, for this opportunity. Thank you.
Thank you, Ms. Sahu. From Pondicherry, the Union Territory, the award goes to Ms. Robina Rahmat Ara, who is MPharm student at Department of Pharmacognosy, Mother Teresa Postgraduate and Research Institute of Health Sciences, Puducherry. Ms. Robina, you're with us. You can share your views.
Yes, sir. Thank you, sir. Myself, Robina Rahmatara, from Mother Teresa Postgraduate.
भी recent तक updation है वो सारा डालना पड़ेगा ना तुम यहाँ जॉब कर रहे ये भी उसको CV में तो डालोगी presently working.
Sir, am I
Hello?
audible?
Yeah. So please unmute your mic.
Am I
Others
audible,
can
sir?
unmute. Yes, you can share your views.
Thank you, sir. Myself, Robina Rahmatara, doing MPharm second year in Mother Teresa Postgraduate and Research Institute of Health Sciences, Puducherry. It is a government of Puducherry institution. I have completed BPharm in the year 2020 in first class with a distinction. I have been awarded the PG Fellowship Award from Tamil Nadu Pharma Welfare Trust, Chennai. I feel really overwhelmed, and I'm very thankful to my principal, Professor Dr. V. Gopal, for nominating me to this award. And thank you, APP, for considering me to this award. Thank you, one and all. Thank you.
Thank you, Ms. Robina. So in the local category, always the hosting college select the best talent candidates in student and faculty level categories from the institute for their unique potential. So in faculty level, local category, among faculty member, we have selected 6 members for their potential. First one is Best Teacher Award from the hosting institute, and this goes to Mrs. AVS Himabindu, who is assistant professor at Department of Pharmaceutics at VIPW Vijayawada. Ms. Himabindu, if you are with us, you can share your views.
Yes, sir. Myself, Hemabindu. I am from Department of Pharmaceutics.
I am very glad for getting this award, sir. I am very much thankful to management of Vijaya Institute of Pharmaceutical Sciences and principal madam for getting this appreciation for me. Thanks a lot once again, sir.
Thank you, Ms. Hima Bindu.
Sir, I would like to add a few lines about her.
Yeah. You can.
Mrs. Hima Bindu, she is a gold medalist in her graduation and a distinction and college topper in her M.Pharm. To add, she is a 7th ranker in AP RSET 2022, the current position. And Mrs. Himabindu, she is a very passionate teacher who is expert at conceptual teaching and always tries to motivate students in all aspects, especially in logical thinking. That's the reason we have selected her for Best Teacher Award. Thank you.
Thank you for selecting Mrs. Hima Bindu for Best Teacher Award.
Thank
So
you, sir.
next
Thank you. Thanks a lot.
Next is Best Researcher Award, and this goes to Mrs. B. Hemalatha, who is Assistant Professor at Department of Pharmaceutics at VIPSW. So congratulations, Mrs. Hemalatha. If you are with us, you can share your views. Or the principal of the institute.
Yeah, Mrs. Hemalata, she is a distinction graduate from KVSR Siddhartha College of Pharmaceutical Sciences, and she has done her MS in pharmaceutics from Naipur, Hyderabad. She is excelled in GPAT and Naipur entrance examinations with All India ranking of 979 and 39 respectively. And she is an avid researcher, and her 4 research publications and 6 review articles till date display her interest in pharmaceutic subject and made her the state first ranker in AP RSET exam in 2022. And she attended various FDPs, participated in seminars, workshops, and conferences, guided M.Pharm students and B.Pharm students. Hence, she has been selected as the best researcher for Best Researcher Award, sir. Thank you.
Congratulations, Mrs. Hemalata.
Thank you very much, sir. I would like to thank my management, Principal Dr. K. Padmalata Madam, for nominating me for Best Researcher Award. I'm very happy to get the Best Researcher Award. Thank you very much, sir.
Congratulations, Hemalata. So our next award is Best Achiever Award, and this award goes to Mrs. Anupma Swati, who is Assistant Professor at Department of Pharmaceutical Analysis at VIPSW. Mrs. Swati, you can share your views if you are with us. Otherwise, principal can endorse.
Mrs. Anupama Swati is also a distinction graduate from Nirmala College of Pharmacy, Mangalagiri, and M.Pharmacy from Vagdevi College of Pharmacy, Gurjala, Guntur. And she is she has a very strong work ethic and multitasker. Apart from teaching, she carries out the departmental and institutional activities with responsibility, and the results are quite evident and beneficial to the students and the institution too. With this basis, we have been she has been selected for Best Achiever Award. Thank you, sir.
Thank you. Thank you. Congratulations, Mrs. Swati. Next award is.
I'm glad.
Next award is. Best Talent Award, and this award goes to Mrs. PMM Nagalakshmi Verma, who is Assistant Professor of Department of Pharmaceutics at VIPSW. Congratulations, Mrs. Verma. If you are with us, you can share your views. Otherwise, principal can endorse.
Mrs. Nagalakshmi was graduated from Sri Padmavati School of Pharmacy, Tirupati, and completed her M.Pharm from Sastra University, Tanjavur. She is an expert in identifying slow learners and advanced learners. She is patient and a steadfast worker committed towards completing the assigned task. Her talent is observed in the clarity of work she presents in her documentation, and hence she has been selected for the Best Talent Award this year. Thank you, sir.
Mrs. Verma, you can share your views.
Yes, sir. A warm and graceful evening for the management and our principal madam, sir, Dr. Padmalata madam. And I'm very much thankful for principal madam for nominating me for this award, sir. And I would like to thank my beloved staff as well as the students who got support for me, sir. Thank you, sir.
Thank you, Mrs. Nagalakshmi Verma.
Thank you, sir.
Next award is Best Pharmacist Award for the Public Service, and this award goes to Dr. N. Parthiba, Assistant Professor, Department of Pharmacy Practice at VIPSW. Congratulations, Dr. Parthiba. If you are with us, you can share your views.
Good evening, sir. Myself Pratibha, Department of Pharmacy Practice, and I would like to thank my management and principal ma'am. I'm very much thankful to principal ma'am. Thank you, sir, for this award.
Okay, congratulations, Dr. Parthiba.
Thank you, sir.
Sir, I would like to add a few points about her.
Yeah, please, you can.
Dr. Pratibha, she has completed her PharmD from Gate School of Pharmacy, Rajahmundry, and she is with us from 2016, and she is with the institution in all levels of head, especially when there is a medical emergency to students. She treats and counsels them at the institution for minor ailments and their problems, and according to the demanding situation, when the accompanies them to the hospital and making it sure that they get the right treatment on time. So with all her social support, we have selected her for this Best Pharmacist Award. Thank you, sir.
Thank you. So the next category award is Best Performer Award, and this goes to Dr. M. Tabitha Saron, who is Assistant Professor at Department of Pharmacy Practice at VIPSW. Congratulations, Dr. Saron. If you are with us, you can share your views.
Good evening, one and all. Myself, Dr. Tabitha, Assistant Professor, Vijay Institute of Pharmaceutical Sciences. I thank one and all, especially my management and my principal ma'am, for electing me and giving me as a Best Performer Award. I thank one and all. Thank you.
Dr. Tabitha too completed her PharmD in 2015 from Gate School of Pharmacy, Rajahmundry. She started her career as clinical pharmacist in Nagarjuna Hospital in 2016, and in 2017 she moved to our institution. She is a certified first aid trainer. She is emphatic, socially responsible, and participated in various health camps along with students, which makes her eligible for this award. So that is the reason we have selected her. Thank you, sir. And I congratulate the newly formed Haldia local branch and the new office bearers of ABB Biotech Division 2022.
Okay.
And also I congratulate all the international and national and my institutional awardees. Thank you, sir.
Yeah. So we have next category award, APP local category student awards. So I will ask here, I will not call the individual students because all are meritous. I have checked the merit for them, but principal as a whole can give the comment at the end. We have selected 5 PharmD students, 4 B.Pharm student, and 1 M.Pharm student. So I will go through all, and then at last I will give opportunity for the principal to highlight their merit. First award goes to the MPhom student, Miss Monica Sarojini, who is MPhom student at VIPW. Congratulations, Miss Sarojini. Second award goes to Miss K. Madhuri Susma Ratnam, PharmD first year student. Congratulations, Miss Ratnam. Next award goes to PharmD second year student, Miss Velangini Data. Congratulations, Miss Data. Next award goes to PharmD third year student, Miss K. Thanmai. Congratulations, Miss Thanmai. Next award goes to PharmD fourth year student, Miss Sumaya Salim. Congratulations, Miss Salim. Next award in PharmD category goes to fifth year student, Miss Tahira Mubin. Congratulations, Miss Mubin, for the achievement. In B-Form category, year first student, Miss B. Harika Ratham is awarded with Best Student Award. Congratulations, Miss Rathan. B-Form second year award goes to Miss N. Jyothika. Congratulations, Miss Jyothika. B-Form third year, the award goes to Miss Payal Ghosh. Congratulations, Miss Ghosh, for the achievement. And B-Form final year, the award goes to Miss K. Manasa. Congratulations, Miss Manasa, for the achievement. Now I will ask the principal of the institute to share a common view about all the meritorious students.
Thank you, sir. On behalf of our institution, Mrs. Vandana Vishnu will share the points about the students. Thank you.
Thank you, sir. Thank you, madam. Sir, the students who have been selected for this Best Student Award, they're all active learners, they're all enthusiastic, and especially some of the students, they are very good at co-curricular and extracurricular activities. And in fact, our students from second B Pharmacy, they have presented papers today. They're inquisitive to learn new things, and especially they are inquisitive to learn about these pharmacy related topics. They keep updated from time to time. And to talk about MPharm student U, Monica Sarojini, she is an all-rounder, sir. She participates in all the institutional activities and helps also us in all administrative activities too, institutional activities. Thank you.
Thank you for endorsing all the meritorious students. So finally, the results of virtual oration competition. So the contest was very tight. We took the average of 4 judges' marks as well as the scientific committee chairman marks. Top scorer is, who is the winner of late Sri Tarsim Jain Memorial Outstanding Oration Award, which will include RS 2000 check, medal and certificate. It goes to Miss Uma from Telangana. She had scored 8.7 as an average from the score of 5 judges. So congratulations, Miss Uma, for getting this award. Our team will contact you regarding the certificate and medal. So if you are with us, you can share your views.
Thank you, sir. Thank you very much. I'm very glad to have this presentation, and I am thankful for you. And I would like to convey my thanks to Dr. Kiranmai madam, who is my mentor in both professionally and personally. And I am very glad to. I want to convey my thanks to my research guide, Dr. R.S. Prakasham Garu, who supported me doing in this work. And thank you very much, sir. Thanks.
Congratulations, Miss Uma.
Thank you, sir.
The runner-up award, which will include a check of 1,000 and medal and certificate, and according to the score, it goes to Miss Jenny, who belongs to Faculty of Pharmacy, Dharam Singh Desai University, Gujarat. She got this award with a score of 8.38, being runner-up. Congratulations, Miss Jenny. If you are with us, you can share your views.
Thank you very much, sir, for bestowing me with this award. And I would like to thank my guide, Dr. B.N. Sohagya, sir, Dean Faculty of Pharmacy, Dharam Singh Desai University. He is my mentor, and I'm overwhelmed to receive this award. Thank you very much, sir.
Okay, thank you. So, third, although third is not associated with the any cash prize, but the fellow will receive a appreciation certificate, and the appreciation certificate for the third prize with a score of 7.7 goes to Dr. Jedha. Congratulations, Dr. Jedha. If you are with us, you can share your views. Okay. So we will conclude here with the announcement of our next conference, APP Indo-Japanese International Conference on Advances and Global Challenges in Pharmaceutical Field, where we will celebrate the World Health Day, and it will be at Wells University, Pallavaram, Chennai on April 9th. Feedback. You can give the feedback after the completion of second day, which will be tomorrow, which is the physical day of the present conference. Today was the online. And I thank you for all. I thank you all for your for giving your time for this award ceremony, as well as this virtual day of the 2-day event. So thank you very much. I conclude here, and now I will invite the Joint Secretary of APP Haryana State Branch, Dr. Ritu Kataria, who will deliver the closing remarks. Dr. Kataria, if you are with us, you can.
Thank you, sir. Good evening, all. With immense pleasure, I'm Dr. Ritu Kataria, Principal, GVM College of Pharmacy, Sonipat, Haryana, is here to present the closing remarks of today's Indo-Malaysian Conference in the commemoration of International Women's Day on theme Global Progress and Upcoming Challenges in Pharmaceutical Sciences, which is hosted by Vijay Institute of Pharmaceutical Sciences for Women, Vijayawada, Andhra Pradesh, in association with Association of Pharmacy Professionals, Andhra State Branch, and APP Malaysian International Branch and APP Molecular Pharma Division. I will start my remarks with the special gratitude for Dr. Rajiv Dhaya, who is founder president of APP. Nowadays, sir is director, School of Pharmacy, the University of West Indies. And my special gratitude is also for Dr. Sunita Daya ma'am, who is General Secretary of APP. And ma'am is nowadays working in the School of Pharmacy, University of Puerto Rico, San John, USA. Also, my gratitude is for Shri B.S. Shri Krishna, who is patron and the secretary, SRK Foundation, Vijayawada, Andhra Pradesh, and for Professor K. Padmalata, who is convener and principal of Vijay Institute of Pharmaceutical Sciences for Women, Vijayawada, Andhra Pradesh, Professor Rajitha Gala, co-convener and vice president, APP, Andhra Pradesh State Branch, Institute of Pharmaceutical Technology, Shri Padmavati Mahila Vishwavidyalaya, Tirupati, Andhra Pradesh. And for Dr. Madhu Gupta, who is a scientific committee co-chairman, and ma'am is working in the School of Pharmaceutical Sciences, Delhi, which is Depsar University nowadays, Pushpa Vihar, New Delhi. For Dr. Gaurav Gupta, who is organizing secretary, national head APP, molecular pharma branch and School of Pharmaceutical Sciences, Jaipur National University, Jaipur, Rajasthan. Conference was inaugurated with the remarks of Dr. Rajiv Dahiya sir, who is founder and president of APP, followed by the welcome speech and opening remarks by Professor S. Shankar, who is retired principal and advisor, ANR College, Kudivada, Andhra Pradesh, and Professor Vetriselvan Subramanian, who is vice president of APP, Malaysian International Branch, and by Dr. Sunita Dahiya ma'am. Inauguration was followed by the extreme knowledgeable lecture session by eminent speakers. Dr. Parmita Basu is Associate Professor, Department of Pharmaceutical and the Biomedical Sciences, Choro College of Pharmacy, New York, USA. And our next speaker was Dr. Shiv Kanya Fuloria, Associate Professor, Department of Pharmaceutical Chemistry, Faculty of Pharmacy, AIMST University, Malaysia. And the session was followed by the intellectual question answer session, which plays a very significant part after every lecture. And everyone thoroughly enjoyed that session also. And there was a wonderful scientific session in which oral presentations and e-posters were presented by several researchers. It was chaired by eminent chairpersons, Dr. Sunita Dahiya ma'am, Professor Somnath De, from St. Paul's College of Pharmacy, Telangana, Professor Piyush Mittal, Department of Pharmacy Practice, Tirthankar Mahavir College of Pharmacy, Tirthankar Mahavir University, Muradabad, Uttar Pradesh, and Dr. Madhu Gupta from Depsar University, New Delhi, Professor Suresh V. Chanupati, Department of Pharmaceutical Analysis, Nalanda College of Pharmacy, Telangana, and Professor Anju Goyal, Department of Pharmaceutical Chemistry, BN Institute of Pharmaceutical Sciences, from Udaipur, Rajasthan. I would like to congratulate all the awardees of today's conference too. And coming to the last part of today's session, in conclusion, I can say that the rejuvenate and the resonating efforts which are put forward by the Association of Pharmacy Professionals and the host institute, that is Vijay Institute of Pharmaceutical Sciences, made this Indo-Malaysian conference a successful event. My special gratitudes are for Dr. Rajiv Dahiya sir, Dr. Sunita Dahiya ma'am, and the special appreciations to the convener, Professor K. Padmalata and her team. Special thanks to the anchoring team as well. Thank you, Dr. Rajiv Dahiya sir, for giving me this opportunity. Thank you, one and all.
Okay. Thank you, Dr. Kataria. So we conclude here, and tomorrow's session will be a physical one, and that will be handled by the principal of the hosting institute.
Sure, sir.
So, okay, madam, bye.
Thank
so
you,
I
sir.
hope today's session was good.
Yes, the
Bye,
whole
sir.
went on successfully without any interruptions, apart from the presentations. During presentations, one or the other hiccups.
Yeah, that was
Everything
some
went
internet issues were there. So
from the candidate side, those problems arise. So other than that, everything went on very nicely on schedule. And I'm very much thankful to Dr. Rajiv Dahiya and Mrs. Sunita Dahiya for making this event successful and giving the opportunity to Vijay Institute of Pharmaceutical Sciences for Women to host the program. Thank you very much, sir.
Okay.
And tomorrow's program, everything is well planned, and I will give you the feedback tomorrow evening.
Okay.
Thank you, sir.
So you have to provide us the photos of the awardees.
Sure, sir. Sure,
Okay.
sir. Yes,
Okay.
sir.
Thank you very much.
Thank you, sir.
Thank you all.